To chair technical session 1, I would like to call upon Dr. Kosala Sirisena from the Department of Environmental Technology to the head table. Now it is over to you, Dr. Sirisena.
Right. Good afternoon, everyone. I would like to welcome you all to the first technical session of the sixth research symposium, annual research symposium of the Faculty of Technology, University of Colombo. In this session, there are three interesting presentations in line. And after each presentation, there will be a 5-minute question and answer session so that you can join with the presenter to ask questions and get clarifications. So let's move on to the first presentation. The first presentation is on identification of molecular expression markers and development of primers for cultivar screening against colorectium musae resistance in banana. The authors are Arunakumara, NVT Jayaprada, N. Thirchelvan, and DMD Costa. The presenting author is Arunakumara.
Good afternoon. I am Aruna. Have you seen this type of banana as market? Banana is having this type of lesions due to post-harvest anthracnose. That is a very tedious diseases caused at the post-harvest stage that reduce the quality of banana at local and international market. To overcome these situations, a screening of varieties against the resistance to this disease is important. Therefore, we started our research based on identification of molecular expression markers and development of primers for variety screening against Colterica. As I explained earlier, the banana is fourth important food crop and second important food crop in the world. Therefore, it has a very high potential to contribute to national economy, GDP, and also to earn more export revenues through export-oriented agribusiness. But significant reduction of significant damage to the banana is caused by these post-harvest diseases. So identification of their resistance, level of susceptibility or tolerance to disease is very important. Therefore, this marker can be used as a very good tool to screen the plants against their resistance to pests and diseases. Understanding the mechanisms of host plant resistance at molecule level is useful towards designing of effective disease management programs against infections of plant pathogens. The screening and identification of genes in resistant banana cultivar would be useful in improving the host plant resistance against anthracnose pathogen. Therefore, the molecular marker proved to be the best alternative for screening against disease resistance. Therefore, we started our objectives and our research with the following objectives. Identification of a set of upregulated genes to infection by Colterica musae. That means we are going to identify some of the genes which express under this infection in a resistant cultivar. Then we select some of the express sequence tags of biotic and abiotic expression molecular markers and their homology function to screen banana cultivar against colotrotricum infection. Then we develop primers for selected expression markers so they can be used for screening based on RT-PCR associated expression profiling. I will explain you the methodology. We have selected the CNE cell that is resistant cultivar against the anthracnose. We infected the well-grown, fully matured banana with spore suspension of C. musae and harvested their peel tissues with time intervals and a total RNA were extracted. Then cDNA were constructed and those cDNA were ligated to the vector and transformed into an electrically competent cell through electroporation to develop the cDNA library. Here you can see the cDNA library. This cDNA library was screened against the resistance of disease by using control and treatment probes. Treatment probe means the probe which developed under the inoculate or infection time or infection mode, and control means without infection. So by doing this differential hybridization, we were able to identify different degrees of disease, mean different level of expressions like up and down regulation. So the probes that mean which treatment probe which we prepare from the infected tissues is showing high signal intensity compared to the control we call as upregulated clones. Those are the genes which express under disease condition. That means that upregulated says about they have particular biological function during the pathogenicity or disease development. The control probe that shows some high signal intensity compared to the treated probes, that means they have some function, that means the gene expression was restricted or down-regulated during pathogenicity. For our further study, we selected upregulated clones. Then as a second experiment, selection of molecule expression marker, annotated gene function of identified expression markers were retrieved from previous literature and KIGO database also we use as a bioinformatics tool. Based on their function, biotic and abiotic expression molecular marker were selected. Then selected markers showed higher tendency of expression during Clostridium musae infection in moderate resistant banana cultivar like CNE. Then, as a third experiment, we developed primers based on selected expression markers. Primer were developed using Primer 3 version 4.1 tool for all these selected markers. The parameters were set as follows: primer size 18 to 20, base pair in length, pair 20 as optimum. Primer, the melting temperature for 57 to 62 degrees centigrade, where 59 were selected for optimum condition to reduce the mean false positive results, and primer GC content were adjusted to 70, where 50 as the optimum and maximum melting temperature defined the differences as 50. These are the parameters we developed. Then as a result from our research, now we are going to discuss the results and discuss more about our findings. These are the results of DNA homology search of the selected cDNA fragment. You can see our different cDNA clones and their homology proteins and homology host, and the accession number we obtained from the EMBL, European Molecular Biology Laboratory, and identity and the E-values. These are the selected clones. You can see from this table. Then possible functions of these identified putative genes were recorded by using available literature. Stress-induced proteins and putative 2C methyl D erythroid 2,4 cytol phosphate synthesis and pectate lyase. Like these are the putative functions of the selected genes. So when you come to this chart, you can see 50% of the identified genes were differentiated genes. That means they can fight fight or they can suppress the growth and development of pathogens. And the 30% stress response related genes, then 10% growth and development related genes, and then 10% protein synthesis, folding, and stabilization related. These are the functions of selected genes. So then, based on the selected express sequence tag, molecular expression markers were developed. Among all these expressed genes in 20 clones, 33 were selected as upregulated expression molecular markers, and 20 upregulated biotic molecular expression markers, and 13 upregulated abiotic stress-induced molecular expression markers were selected for the analysis. Key biotic selection markers and their functions are varies, and germline like protein, they have very good response against to infection of diseases. And endokinase and ubiquitin conjugative enzymes are having a very good immunoresponse during fungal infestations. And also chitinase, protein has a differentiated genes that can react against the fungal development. So they can digest the fungal mycelium to avoid the further infection into the plant tissue. Cellulase synthase and sucrose synthase are other expression markers. They actively in vascular tissues reported to support for the production of cellulose necessary for thickened secondary cell wall during the pathogenicity. Other significant markers like upregulation of the lignin biosynthesis, ethylene biosynthesis, and chitinase synthesis associated immune response activities according to the literature. Key abiotic selection markers and their functions are also diverse. We have 13 abiotic markers, 6 abiotic stress resistance such as cold, drought, and other, and like salinity, and methylthionine-like protein expression markers in MUSA, in 706 expressed for plant immune against higher concentration of heavy metal exposure. The other expressions marker were associated with plant protein protection from oxidative, the plant protection, cell protection from oxidative burst. That means they can react with the reactive oxygen species and they can neutralize and avoid the oxidative burst. Otherwise, cells are burst and ultimately cellular collapse can be resulted, leading to death of the plant. And these are the information, so the data which we collected as a selected biotic expression molecular marker through our seeding library construction, and their respective developed primers can be seen through these tables as our findings. And further to you can see those putative, the express sequence tag, and they are possible the primers that we can use to evaluate the resistance levels of plants against to anthracnose pathogen. And these are also the same results which we obtained under the biotic expression molecular markers. And this table shows you about the abiotic expression molecular markers and their respective developed primers, which you can use for a varietal screening to identify which variety showing resistance against to the anthracnose, and those cultivar can be used for crop improvement or further plant breeding processes, and also screening the nursery plant before come into the field. Likewise, finally we can conclude the present study identified 20 upregulated cDNA clones by differential hybridization of a cDNA library for banana infected with Symposium, and selected upregulated cDNA clones containing a transcripts are responsible for defense, stress response, growth and development, protein synthesis, protein folding and stabilization, like in that as mentioned in the chart. And 20 selected molecular expression markers were associated with biotic responses for choleric mucus infection resistance, and there 13 were associated with upregulated abiotic responses. cDNA primers for all the selected markers were designed as well. And further, we can use these upregulated clones for the further biotic molecular expression markers may be particularly important in choleric mucus infection resistance in banana cultivars and identification. These selected expression markers and primer will be useful for scientists to design coloricum user infection and to see screen musa cultivars against coloricum infection resistance based on gene expression profiling. These are the literature which we use for our study during this time. It will be difficult to dealing with molecular level study. That's what we have to put our effort to bioinformatics tools to develop further research and improve our research and development activities. I would like to acknowledge National Research Council and Faculty of Agriculture of Peradenian, Department of Agriculture Technology, Faculty of Technology, University of Colombo. Thank you very much.
Right. Thank you very much for this interesting presentation on your research. And now it's the time for question and answers. If you have any question or if you would like to get any clarification, now it's the time for you to ask questions from the presenter. We have 26 online participants. You also can join with this session if you have any question to ask from the presenter. Until we get questions from the audience, I would like to ask a question from Dr. Aruna. According to the explanation, I got the feeling that the gene expression can be influenced by the environmental factors as well, like the storage conditions and all these things. So, the markers that you develop, how can you apply in different storage conditions or environmental conditions?
Actually, we have selected these molecule expression markers based on up and down regulation. Basically, under the infection, the genes are expressed as upregulation. Even we have selected some biotic expression marker and abiotic. Therefore, as you mentioned, environment condition like drought, salinity, or even temperature, those things also we have separate set of genes we have selected for biotic and abiotic condition. Therefore, these abiotic molecular expression markers can be used to identify the environmental parameter where the plants are or stored products or banana exposing. That's what basically we try to screen the plants against resistance to the biotic agent, because this is infected at field level, but the symptoms appear during post-harvest stage. That's what we have selected two different type of these markers for a biotic condition where the pathogens are infected. Other things are for environment parameters. We can use both for two different scenarios.
Right. Thank you. Any questions from the audience?
Dr. Aruna, I'm having a small question about the infection stage. So, as I understand, this is just a clarification. You said that the infection will be even before the ripening of the bananas.
Yes.
Is that so? So, your markers can be used even before the ripening of the bananas?
Yeah, actually, this condition is like a latent infection. That's what we cover this banana before we harvested. Because you know the anthracnose is caused by coltricum as an aerial infection. That means they come from the air and deposit on a peel tissue. They stagnated there due to latex. Because latex is not good for a pathogen because that is like a fungicide. Once they come to the physiologically matured stage and they started to ethylene production and ripening, they initiated. That's what basically this pathogenicity or a disease development is basically resulted during post-harvest stage. During a pre-harvest stage, there's no any symptoms or any sort of, just we can see some type, that's what we have a set of genes which collected from before inoculation and after inoculation. That's what it's a full of genes, my dear. That the infection during field level, we cover the banana with perforated bags to avoid the environmental contamination. Then we any symptom were developed, the infection from the field level.
Right, thank you. Any questions from the audience? Right, so it seems like we don't have any questions from the audience. So thank you again for your nice presentation.
Thank you very much.
Thank you very much. So let's move on to the second presentation in our line. Our second presentation is on antimicrobial activity of selected traditional and new improved rice varieties of Sri Lanka. The authors are A.T. Liyanage, D.U. Rajawardana, W.P.K.M. Abesekara, and W.K.S.M. Abesekara. The presenting author is A.T. Liyanage.
Good afternoon, all of you. I am Takshila Liyanage. Today I am here to present the research of antimicrobial activity of selected traditional and new improved rice varieties of Sri Lanka. These are the subtopics that I am going to talk about. Let's look at the introduction about rice. Rice is the one of the world's most extensively cultivated cereal and the staple food for over half of the world population. Traditional and new improved rice are the two main cultivated rice varieties of Sri Lanka. The improved rice are mostly cultivated and mostly consumed, and the traditional rice varieties are indigenous and developed using natural selection. Except the whole rice grain, the rice bran is proven as the most antioxidant, anti-cancer, anti-diabetic properties rich portion by many researchers throughout the globe. It contains a number of bioactive compounds such as ferulic acid, linoleic acid, alpha-tocopherol, salicylic acid, and etc. When we look at the reason for conducting this research was the antimicrobial activity of traditional and new improved rice varieties of Sri Lanka is still limited. Therefore, the objective of this present study is to evaluate the antimicrobial properties of selected traditional and new improved rice varieties of Sri Lanka. Next, we move to talk about the methodology. The 10 rice samples were provided by Rice Research Institute of Bathalegoda and Ambalanthota. These are the 5 traditional rice varieties that I have used for this study. Sudu Heenati, Koda Heenati, Tatuvi, Ratu Heenati, and Elvi. These are the new improved rice varieties: BG300, BG352, BG360, AT373, and AT362. For the bran extraction preparation, separated rice bran from the dehulled rice samples were extracted with 70% ethanol by using orbital shaker. Then, solvents were centrifuged for 10 minutes and the remaining ethanol were evaporated by using rotary operation process. After that, the samples were freeze-dried and bran extract were collected into the Eppendorf tubes. Here I mentioned the test microorganisms that used for this study. There were 5 bacterial species and 1 fungal species. All of them were pathogenic microorganisms. For the evaluation of antimicrobial activity, agar well diffusion assay were carried out. There, the wells were punched on the inoculated agar plates by using a sterile cork bore. The diameter of cork bore was 8 mm. Then wells were filled up 200 microliters of rice bran extract separately and the plates were incubated at 30 Celsius degrees. After that, the diameter of inhibition zones were measured. For the statistical analysis of this research, two-way ANOVA test and the statistical software Minitab 17 were used. Let's talk about the result and discussion. At the 3 mg per milliliter concentration, none of the new improved rice varieties had antimicrobial activity for any of the tested microorganisms, whereas except LV, all other traditional rice varieties had antimicrobial activity only against Bacillus cereus, and for other organisms there were no activity. However, among rice varieties, there was significant difference for antimicrobial activity of Bacillus cereus. The highest activity was shown by Tatuvi and the lowest activity shown by the Godahinadi rice extract. At the 5 mg per milliliter concentration, the rice varieties showed the antimicrobial activity for Bacillus cereus and Staphylococcus aureus only. Here, except LV, all other traditional rice varieties had antimicrobial activity, and among the new improved rice varieties, only 8362 variety had shown the antimicrobial activity. In general, Tatuvi showed the highest antimicrobial activity and 8362 showed the lowest activity against Bacillus cereus and Staphylococcus aureus. These are the actual photographs of assay plates with inhibition zones at 5 mg per milliliter concentration. At the 10 mg per milliliter concentration, the rice variety showed antimicrobial activity for Bacillus cereus, Staphylococcus aureus, and Enterococcus faecalis. Here, Tatuvi showed the highest antimicrobial activity against Staphylococcus aureus, and Sudhuhinati showed the highest activity against Bacillus cereus and Enterococcus faecalis. In general, 8362 rice variety showed the lowest activity against these three organisms. These are the assay plate photographs of bran extract at 10 mg per milliliter concentration. At the 15 mg per milliliter concentration, the rice variety showed antimicrobial activity for Bacillus cereus, Staphylococcus aureus, and Enterococcus faecalis. In general, Tatuvi and Sudhuhinati showed highest antimicrobial activity against these three organisms. Ratuhinati showed the lowest activity against Bacillus cereus, and Godahinati showed the lowest activity for Enterococcus faecalis. These are the actual photographs of inhibition at 15 mg per milliliter concentration. The standard drug used for this study was gentamicin in concentration of 10 microgram per milliliter. Compared to the standard drug, rice bran extract showed the moderate activity. As the conclusion, among investigated rice varieties, Sudhuhinati, Godahinati, Ratuhinati, Tatuvi, and 8362 varieties showed the antimicrobial activity. That rice varieties showed antimicrobial activity only against Bacillus cereus, Staphylococcus aureus, and Enterococcus faecalis. Among investigated traditional rice varieties, Sudhuhinati and Tatuvi showed significantly high activity than other two varieties. Except 8362, none of the new improved rice varieties showed the antimicrobial activity against any of the tested microorganisms. Also, all rice varieties which showed antimicrobial activity consist of red rice bran. The results strongly indicate that traditional rice varieties have significantly high antimicrobial activity than new improved rice varieties. And also, red rice bran is a potential source of antimicrobial activity for further research. This study was conducted at Industrial Technology Institute. Therefore, I would like to acknowledge this institute to provide all the facilities to carry out this research. These are the some of the reference I have used during my research. Finally, I would like to thank you all, and if you have any question, I will be happy to answer.
Right. Thank you very much for your excellent presentation and your very interesting research work. So as the presenter mentioned, it's time for question and answers. If you have any question, please go ahead.
Thank you very much for the nice presentation. Just I want to clarify something. When you do the dual plate assay, what did you do? That mean you have spread the brain solution first and add the concentrated microorganism to the spot, or in other way around? What is that mean? Just to clarify that one. When we do, can you understand my question?
Sorry, sir, I didn't get the question.
Can you explain the procedure? That mean when you do the ECA dual plate assay, first you spread the
First, I applied the bacterial inoculum solution on the agar plates by using a cotton swab. After that, I punched the wells on the agar medium. After that, add the bran extract for the wells.
That the concentration of that brain extract.
Sorry, sir?
You mentioned some concentration of something that you had?
Yes, sir. I studied 3 concentration levels.
Of bacterium or
no, sir. Bran extract.
Brain extract
Concentrations levels of bran extract.
What the concentration? That mean about what they are? Just to clarify, bran extract concentration mean like just I am telling like microbial concentration like colony forming unit, spore per ml. Like what is that standard that you use as a concentration? Just for understanding.
McFarland stand, sir. Okay. 0.5 concentration, McFarland stand.
Okay. So first you spread and incubated overnight to get the culture. Culture, then you puncture the wells and put the concentrated brain extract and observe the diameter of the area where that is having some control or clear zone. That the way.
After applying the inoculum, I already add the bran extract. After that, all the plates incubate.
Okay. Then you measure the diameter of the clear zone.
Yes, sir.
Okay. Thank you.
Thank you, sir.
Any other questions? Yes.
Thank you, Takshira. Hello.
Yes, madam.
That was a nice presentation. I'm having a small question. Can you explain about the basis of selection of those 5 types of bacteria and the fungal species?
Madam, all of these 6 microorganisms are pathogenic microorganisms. Because of that, I use these types of bacterial and fungal species. then also the availability of the microorganisms in that institute. That is the reason for I use these types of microorganisms for this study.
So what can I understand is you wanted to prove the enteroprotection or the gut protection given by the traditional rice varieties over the new improved rice varieties, right?
Sorry, madam?
You wanted to prove the enteroprotection given by the traditional rice varieties compared to the new improved rice varieties.
Yes, madam.
Okay, that should have emphasized in your study because that was the most important point in your study. It's a small suggestion,
madam.
right? Thank you.
Thank you, madam.
Right, thank you. And I also have a small question. Actually, probably this could be the higher end of your research. How can we determine the dosage which is suitable for consumption? Like when we take antibiotics, we know we take specific dosages depending on sometimes depending on age and likewise. So in this case, how can we determine the dosage and will there be any adverse effects like antimicrobial resistance, like we have antibiotic resistance if we use excess amounts of these rice varieties?
So could you please?
I was asking when we take excess amounts of antibiotics, antibiotic resistance will be developed. So in this case,
yes.
If we, how can we determine the dosage, whether there are any limit that we can't consume more than that? After that, there'll be like antimicrobial resistance, something like that.
Yes, sir. For you, instead of using the chemical synthesis antibiotics, and these are the natural things. We can use the natural antibiotic compounds for that.
I asked this question just for the curiosity. Yes, Dr. Manchal,
yes, Dr. Kausal, can I answer to that question? Actually,
yes,
it's
please.
a nice question, good one. Actually, that we need further studies to, answer to that question. We need like clinical level studies. And also now compared to the standard drugs, the activity is moderate. So there are so many actually thousands of traditional rice varieties. Our plan is to first screen the varieties and identify the most potential, highly potential varieties. Then as you said, we will plan like clinical level studies so that we can decide the doses and so on. Thank you, Dr. Kausal.
Yeah.
Yes.
Thank you very much for the nice presentation. Let's move on to the last presentation in this technical session. Our last presentation is on anti-glycation and glycation reversing activity of 3 medicinal plants which are well known for the management of diabetes mellitus in traditional medicine. The authors will be C.M. Piris, W.K.S.M. Abesekara, S.B. Navaratna, and A.P. Henegamage. The presenting author is C.M. Piris.
Good afternoon to you all. I am Mahishika Piris from ULS University. Today I am going to share my findings with you on anti-glycation and glycation reversing activity of Costa species, Cocconia grandis and Gymnema sylvester freeze-dried leaves. I registered for the split-side PhD in Faculty of Graduate Studies, University of Sri Jayawardhanapura, collaboratively doing with Curtin University, Western Australia, and these experiments were some parts of my PhD study. My PhD research granted by the AHEAD grant. In here you can see the lineup of my presentation, and I am going to discuss all the topics in here throughout my presentation. I hope you may be aware about the diabetic mellitus. It is a metabolic disorder that prevails worldwide in alarming rate. According to the WHO, they predicted more than 300 million people will be suffered from diabetics in 2025. Meanwhile, there is a gaining potential for herbal medicine to treat the diabetics in all over the world. Let's move to my research problem. The disease is diabetic mellitus. It can be short-term, long-term, or combining with other disorders. Currently, chemically synthesized drugs were used to control the diabetics, for an example, metformin, acarbose. However, their cost is high as well as they have side effects. Therefore, it is better to find a replacement for these synthetics with herbal medicine because presently naturals have high demand in consumption. You all know that Sri Lanka is blessed with variety of medicinal plants. However, still few medicinal plants have been evaluated for anti-glycation activity. So my target is to promote herbal extract for the development of nutraceuticals, supplements which have the anti-glycation activity. In addition to that, you all know the moisture of herbals can promote the undesirable enzymatic reactions as well as the microbial contaminations. Therefore, we can't keep fresh materials for a longer time periods. To eliminate the moisture or else to obtain the powdered products, then we can apply the various drying techniques, namely shade drying, freeze drying, vacuum drying, oven drying, sun drying, etc. However, the stability and the composition of bioactive compounds can be affected by the application of drying technique. In this study, I selected freeze drying method because it is promising to preserve the bioactive heat labile compounds. Let's move to my objectives. My objectives are to determine the anti-glycation and glycation reversing activity of freeze-dried leaves of selected medicinal plants and to select the most effective plant species among them for the anti-glycation and glycation reversing activity. Now I am going to explain my methodology. First, I selected the plant materials. 3 different medicinal plant leaves were selected, namely Kovakka, Tebu, Maspatha, and authenticated them from the National Herbarium Peradeniya. Specially, I selected these leaves due to their high availability, non-toxic, edible, not seasonal, year-round we can find them, not harm to the whole plant, environmental friendly, ease of cultivating. In traditional medicine, they recognize with anti-glycation activity. Here you can see the classification of selected species starting from Cocconia grandis, common name is Kovakka, Tebu, Costa species, Gymnema sylvester, common name is Muscat. Then the plant leaves were well washed and prepared for the freeze drying process. The leaves were freeze dried until they get the constant wet. After the freeze-drying process, leaves were ground to obtain the powder, and extraction was done by using maceration method to protect the heat labile compounds using 95% ethanol 1 to 10% ratio at the room temperature. Excess solvent evaporated with using rotary evaporator, and crudes were subjected to determine the anti-glycation and glycation reversing activity with using bovine serum albumin glucose model. Here the routine used as a positive control, and the fluorescence was measured with use of fluorescence 96 well microplate reader. Data statistically analyzed using Minitab 17 software. Let me explain you the results and discussion. In this slide, you can see the freeze-dried leaves of selected species before and after the grinding process. According to the table 1, final moisture content of all plant leaves were ranged in between 5% to 6%. The lowest drying time and the highest drying yield were obtained by the Chimnima sylvester. The extract yield was high in both Costa species and Chimnima sylvester. Let me explain you about the anti-glycation activity of selected leaf extract. All leaf extract show the dose-dependent anti-glycation activity. Among the species, freeze-dried Costa species leaf extract showed the significant anti-glycation activity compared to the other two species. Similar to the anti-glycation activity, the glycation reversing activity also the highest in Costa species, and if we increase the concentration simultaneously, glycation reversing activity also increased. Here you can see the IC50 values and EC50 values of selected species. There was a significant difference in both anti-glycation IC50 values and glycation reversing EC50 values with the plant extracts. The lowest IC50 values and EC50 values shown by the Costa species leaf extract. However, the Costa species values approximately 5-fold higher than the positive control routine. Finally, I would like to conclude my findings. All 3 freeze-dried leaves of Costa species, Cocconia grandis and Chimnima sylvester ethanolic extract showed the higher anti-glycation and glycation reversing activity. Out of the selected species, freeze-dried leaves of Costa species was the most effective for inhibiting protein glycation and the glycation reversing activity. Here you can see my references. Finally, I would like to acknowledge all of those who helped me in the Department of Agricultural Technology, Faculty of Technology, University of Colombo to conduct this experiment. Thank you.
Right, thank you very much for this excellent presentation. Now it's time for questions. Until we get questions from the audience, I have a small question. What is the importance of using a freeze drying method rather than the conventional methods that we use for processing tea and things like that? I'm asking this question because sometimes there might be adverse effects on, probably there might be some adverse effects on using extreme conditions in freeze drying, especially alternation of chemical compounds and things like that. So what was the importance of using freeze drying method for this, and the cost effectiveness compared to the conventional processing methods like tea?
Preserving the bioactive compounds. So here actually I am concerning about the anti-glycation and glycation activity, and again the bioactive compounds. So therefore actually I used the freeze drying method to preserve the bioactive compounds. So that is why I used the freeze drying method. And other than that, if we consider about the cost, the freeze drying is somewhat cost method, but my concern is to get the maximum bioactive compounds which are having the anti-glycation and glycation activity for the developing of supplements. So that's why I use the freeze drying method, and again it can preserve the heat labile compounds because we are not providing high temperature in the freeze drying method. And with the crystallization process, we can preserve more bioactive compounds using that method. That's why I selected freeze drying method.
Right, thank you.
Yeah, I have a question that you mentioned some bovine serum albumin-based method for detection of these activity of the compound or plant extract. Can you just explain me a little bit about that mechanism? I mean, there will be some reaction. Just to clarification, what are you going to measure with the help of this plate reader? Just for the understanding.
Yeah, sure. Actually, I used the bovine serum albumin method with the glucose model actually. So this is, I determined the anti-glycation and glycation reversing effect. So actually in our body, the reaction, there was a reaction in between the reducing sugars and the free amino acids. So with the reaction of these reducing sugars and the free amino acids, it can produce the advanced glycated end products. So these glycated end products are promoting these diabetic complications. That is the main reason. So this is the same mechanism we tried with this bovine serum albumin glucose model. The glucose is the sugar, and bovine serum albumin, it provides the amino acids. And with the combinations of, so I actually tested with my extract how they can inhibit this protein glycation. Actually, it is like a Maillard reaction. It's a non-enzymatic reactions. So it's produced some heterogeneous mixture. They are called advanced glycation end products. So that's why actually I tested with this in vitro model using the same mechanism in vitro and tested the fluorescence advanced glycation end products with checking the fluorescence values using 96 micro well plate reader.
Specific that, like say, according to my knowledge, by using this plate reader, we can measure some compound with their wavelength or some fluorescent emission. Are there any end product that you are targeting to measure? As during this heterogeneous compound, there is a particular product that has
the fructosamine. So there are different end products we can generate. We can actually generating during this process. So specifically, we can't say this type is we can find in that reaction. It's actually collection of the mixture.
That means you are measuring some fluorescence.
So then I check the the fluorescence advanced glycated end products. There are fluorescence ones and the non-fluorescence ones. So with the fluorescence, I checked the fluorescence glycation end products.
Right. Thank you very much. Any other questions? Right. It looks like there are no more questions, and that concludes the first technical session of this symposium. And I would like to thank all three presenters for presenting their research, and I would like to thank the audience as well for making this session very interactive. Thank you very much.
Marks the end of technical session 1. To start the technical session 2, I would like to call Mr. Thilini Jayaprada from the Department of Agricultural Technology to chair the session. It is over to you, Ms. Jayaprada.
So this is the technical session number 2, and there are going to be total 3 presentations. So we can move on to the first presentation. The title is The Levels of Nitrite in Locally Produced Fresh and Preserved Asian Black Tiger Prawn Penaeus monodon, authored by E.V. Piris, J.J. Vevalvala and S.R. Gunatilaka. The paper is going to present by Ms. E.V. Piris.
Good day, ladies and gentlemen. My name is Isini Piris, and I am here to present my research work on the analysis of the levels of nitrite in locally produced, fresh and preserved Asian black tiger prawn, the Penaeus monodon. This research work was carried out as a collaborative project between the Department of Agricultural Technology, University of Colombo, and the College of Chemical Sciences, Institute of Chemistry, Ceylon. The black tiger prawn, the giant tiger prawn, or Asian black tiger shrimp, is a marine crustacean that is widely caught and farmed for food. It is, in today's seafood market, the P. monodon is considered a top quality niche consumable. And in Sri Lanka and all around the world, the prawns are popular as a food source and are of great commercial significance due to their very high nutritional value. Sodium nitrite is an inorganic salt of sodium containing nitrite as the counterion. It serves as an antimicrobial food preservative, an antidote to cyanide poisoning, a food antioxidant and antihypertensive agent, and also a poison. Sodium nitrite is also a food additive approved by the USDA and is very widely used in curing meat and meat products, and it serves as a color fixative and prevents the growth of bacteria such as Clostridium botulinum. It is also a strong reducing agent which includes antibacterial properties, but a higher than necessary amount of sodium nitrite when consumed may give rise to a condition known as methinoglobinemia, which is a peculiar and potentially fatal disease. It can also cause cancers in the bladder and has been identified by the IARC as a group 2A human carcinogen. Sometimes seafood or any other meat products can't be immediately supplied to the market when they are processed. Sodium nitrite is commonly used as a preservative in commercially available fresh and frozen black tiger prawns, and the amount of nitrite used is sometimes not regulated. So it is hypothesized that due to the increased addition of nitrite for longer storage periods and unregulated addition of sodium nitrite, there may be excessive levels of nitrite causing toxicity to consumers. To determine the levels of nitrites in these black tiger prawns, P. monodon samples were collected from various sampling sites, and the grease spectrophotometric analysis method was used to find out whether the levels of nitrite are above or below the permitted standards. The main principle of the method, the grease spectrophotometric analysis method, involves acidic conditions where the nitride reacts with an aniline derivative such as sulfanilamide to form a diazonium cation, as you can see in the slide here, which then reacts with a coupling agent such as NED in an azocoupling reaction to form a pink-red water-soluble azodine. The nitride detection and quantification by the grease assay is a two-step process where first, initially an aliquot of the sample containing the nitride ion is added to an acidic solution of sulfanilamide, and afterwards it forms the diazonium salt, and then on the addition of NED, the azo dye is generated. Once the azo dye is formed, it is spontaneously protonated to yield the species that is responsible for the pink-red color of the grease assay. Method development involved 6 major steps. First, samples were collected in triplicates from 9 different sampling sites, and then they were stored at minus 20 degrees Celsius prior to the analysis. The samples were then homogenized and extracted using DI water and borax solution. Following the extraction, protein precipitation was carried out using the Curies reagents to minimize interferences that may occur during the analysis. Afterwards, sulfanilamide and NED were added to induce the formation of the diazonium compound, following which the absorbance of the colored solutions were measured using UV visible spectrophotometry. Method validation consisted of the determination of detection limits, the limit of detection and the limit of quantification. Evaluation of the metric effects and matrix effect and recovery tests were also carried out. A neat calibration curve was first plotted for a standard series of nitride solutions composed of known amounts of sodium nitrite and DI water, and the LOQ and LOD were calculated using the signal to noise ratios. For matrix evaluation, a matrix calibration curve was plotted following the neat calibration using a series of sample extracts instead of the DI water, spiked with the same nitride concentrations as in the neat calibration curve. The matrix effect was determined by comparing the gradients of the neat calibration curve and the matrix calibration curve. Spike recovery tests were also carried out to determine the efficacy of the extraction procedure. A high spike and a low spike recovery test was carried out by spiking the samples with the relevant concentrations, and the extraction procedure was carried out followed by the sample analysis. The method detection limits were found as follows. The LOD was found to be 7 micrograms per kilogram, and the LOQ was found to be 23.3 micrograms per kilogram, and these limits were determined using the standard deviation of the neat calibration curve by the linear least square method. The average matrix effect, as you can see, was calculated to be 6.25%, which is insignificant, and therefore no matrix calibration was required for any of the sample analysis, and a direct analysis was possible. The spike recovery values were found to be 20 ppm. Spike recovery values were found to be 29.22 for the low spike, which was carried out with a 5 ppm spike value, and the high spike recovery was found to be 122.96% with the spike value at 20 ppm. 27 samples were analyzed, and this slide displays the average nitride levels in all of them in micrograms per kilogram. The highest levels of nitride were found in the Nillaveli samples and the lowest were found in the Chilau samples, but even so, they show no significant differences. Finally, to conclude, The detection limits were the LOD was 7.0 micrograms per kilograms and the LOQ was found to be 23.3 micrograms per kilograms. And the method is determined to be sensitive enough and accurate and reproducible. The average matrix effect obtained was 6.25%. Therefore, matrix effect is insignificant. Therefore, a direct analysis is possible. High spike recovery was found to be 122.96% and low spike recovery was found to be 29.22%, which determines that the method developed is accurate and precise. Therefore, our method is reliable. All samples contained nitrite levels that were well below the standard permissible limit of 200 ppm for nitrite concentration in food as regulated in accordance with the Food and Drug Administration standards. From this preliminary study, we can conclude that prawns acquired from all the sampling sites were safe for consumption. However, for an overall conclusion, a broader study is required to be carried out. Finally, I would like to acknowledge the Institute of Chemistry Ceylon and the Faculty of Technology, University of Colombo for providing the facilities and the space to carry out this research project and conclude it with success. Thank you, and I am open to answer any questions.
Thank you, Ms. Piris. Now the presentation is open for discussion. Any questions from the audience?
That means you are going to test the level of nitrate in the homogeneous sample. In your procedure, you mentioned you are going to precipitate protein rather than precipitating entire sap or the entire homogeneate. Can you explain what the reason for precipitation of the protein in the lines of analysis? Can you understand the question?
Hello?
Did you get the point?
Yes, sir, am I audible?
Yeah, really.
Right. So actually, it's an analysis of the nitrite ions. So the purpose of precipitating the proteins was because if the proteins are retained in the solution, it causes a turbidity. Since we are carrying out a spectrophotometric analysis, to get rid of that turbidity only, we have to precipitate the proteins specifically.
That you better get the solution to analyze. It contain entire homogeneate, but according to your clarification, that means to prevent some residues effects. Is that the answer?
Sorry, sir?
You are going to use the protein precipitate to eliminate some residues.
Yes, it is basically to get rid of any turbidity, because since we are carrying out a spectrophotometric analysis, our final solution must be clear. So to get the clear solution only, we have to precipitate the proteins.
That means you are actually really precipitating those say residues to get the clear solution. You analyze the clear solution with the help of the spectrophotometry.
Yes.
Okay, I've got the point. Thank you.
You're welcome, sir.
Okay, Ms. Piris, I have one question. So I think this sodium nitrite is giving chronic toxicity. That means the symptoms will give after exposure to small amounts for a longer time of period, right? So are there any cases that have been recorded in Sri Lanka during any point of time?
According to literature in Sri Lanka, not so much. I have not come across any single reported case. But in certain other countries, there have been reported cases of methylglobinemia and cancers caused due to nitrite poisoning.
Okay. I have one other question. So do you have any recommendations that we need to be aware of to get rid of the sodium nitrite from the meat products or food products at this stage of consumption or usage?
Sorry, madam, the question is not very clear. Could you repeat that, please?
So are there any methods that we can get rid of the sodium nitrite from food products at this stage of consumption?
At the stage of consumption, madam, it's like this: if it is fresh meat or seafood, best would be to wash it well, because sodium nitrite is water soluble. So I think washing it properly would help us to get rid of excessive residues of sodium nitrite. But if it is canned or processed meat products, there is really not like a particular way that we could get, like, there's nothing that we can actually do to reduce the amount of sodium nitrite in it. But then again, mostly in canned foods and processed foods, they normally indicate the levels of nitrite that are included in the food product. So my analysis was carried out for fresh seafood or the frozen ones that are for sale in the supermarkets, not really canned and processed. So for those types of food only, the sodium nitrite is not that regulated. The amount they add, they just add without much proper regulation. So in those cases, I think washing the food before consumption, washing the fresh seafood or meat before consumption is pretty much the only thing that we can do.
Okay, thank you, Ms. Piris, for your informative presentation and answering the questions. Any questions from the audience?
Yeah, I do have one question. How about the interference from fat?
Sorry, madam?
How about the interference from fat? Because it contains fat, food contains fats. So how do you overcome that problem?
Madam, the procedure, sample preparation procedure involves protein precipitation, but also other steps like centrifugation, and we added potassium ferrocyanide and sodium hydroxide, and from that we precipitated all materials that can actually interfere. So the final analyte that we obtained was a completely clear solution, which could be analyzed using our spectrophotometric method.
Yeah, but the foods containing more fat, I think initial stage you have to defat it.
Sorry, madam?
Foods containing more fat, the interferences are also there. I think the one that you can do is you have to first defat your sample.
It's like this, madam. The analysis procedure also involved the recovery studies that we carried out. So the recovery studies was done to see whether, what we did with the recovery studies was we spiked our sample with a known amount of nitrite, and we carried out the extraction procedure to see if that actual nitrite was extracted later on, like the final analyte actually contained the nitrite that we spiked initially. Yeah. So the recovery values were well within reliable range. So I think that we have minimized interferences with our sample preparation procedure.
Yes, thank you. Thank you.
Thank you, Ms. Piris. I think we can move on to the second presentation. The title is Assessing Greenhouse Gas Emission of Low Country Orthodox Black Tea Manufacturing Process in Sri Lanka, authored by MDND Pereira, AWDR Kalpani, CM Navaratna, GY Jayasinghe, and SB Navaratna. The paper is going to present by Ms. MDND Pereira.
Good afternoon. I'm Nimeshika Pereira from the Department of Agricultural Technology. Our study was conducted on assessing greenhouse gas emission of low-country orthodox black tea manufacturing process in Sri Lanka. Here is the flow of our presentation. The brand of Ceylon tea, a quality production from Sri Lanka. As we all know, with their famous and greatest brand name, we are in the 4th place among tea producing countries while being 3rd at the export market. In both production and export, India and China has taken first and second places consequently. But with the delicious flavor and smell, the Ceylon tea have been placed as the best orthodox supplier for the black tea market. When considering about the black tea production by elevation in Sri Lanka, the highest contribution has been given by the low-grown factories. So in here, the graph indicates that during the last decade, the low country contribution has been continued around 60 to 65% from the total production, while keeping a significant gap with the other two regions. Further, with market trends for being precision and sustainable, reduction of emissions and building resilience must be practiced in the industry level. When considering about the Sri Lankan tea industry, factory level emission and energy use is in a significant level according to the previous studies and comparisons. Therefore, for conducting life cycle assessment, all the steps should be taken into the inventory for assessing the greenhouse gas emissions. On the other hand, acting on carbon could produce new commercialism points for products and services supplied to the market. It will assist to sell products with high environmental standards at a premium price. And also, several studies have been already conducted for assessing the carbon footprint of tea process in Sri Lanka. But recommendations have not been based on the elevation and process steps. Therefore, carbon footprint is an essential part in low country tea process as they are the highest black tea producers in Sri Lanka. With these scenarios, our study was conducted with these research objectives. The first one was to analyze the specific energy consumption in low country tea factories. Second was to calculate greenhouse gas emissions of low country tea manufacturing factories at each processing stage due to the electricity consumption. And third objective was to analyze greenhouse gas emissions in use of different energy sources for manufacturing black tea. And finally, to determine the total greenhouse gas emissions in the low country orthodox black tea production. Materials and methods. The study area was low country region and purposely 3 factories were selected according to these criteria, especially the location, average annual rainfall, average annual temperature, and finally the annual average production, which is more than 3 million kilogram per year, were considered for the selection of 3 tea factories. Here is the process flow that we considered for the life cycle assessment, and inventory data was gathered following these steps mainly. At each and every step, the materials, type of energy, and the amount of energy used were taken into the account. In data collection and analysis, electricity base and production details were collected. In case of electricity base, especially the total consumption, peak, off-peak, day consumption, and apparent power were considered. For the production details, made tea, refuse tea, and fresh leaves were considered. For the secondary data, specific energy consumption benchmarks were considered, which were gathered from the Sustainable Energy Authority. Specific energy consumption was calculated according to the previous studies and SCA benchmarks. In calculations and data analysis, 1 kilogram of made tea was concerned as the functional unit for all the steps. The inventory of data and calculations for processing stage was conducted according to the taken conversion factors from the previous studies and the SCA benchmarks. Results and discussions. For the first objective of specific energy consumption, firstly, we calculated the electricity use in the factories. According to that, the phase of day, the highest energy consumption was recorded in all three factories. And also, the off-peak consumption is higher than the peak consumption. But most of the factory operations have been taken at the day phase. In specific energy consumption in low quantity factories, the lowest specific energy consumption was recorded in factory 2 and it is nearly achieved the best achievement which has been already given by the SEA and also according to the benchmark of Sustainable Energy Authority, it was 0.82 and according to our values, it has been already achieved by two factories. When we consider about the specific electrical energy consumption in low quantity factories by stepwise, the highest energy consumption was recorded at the withering stage following by drying stage. And also when we consider about the total energy consumption, it was around 0.80 and it is nearly equal for the value which was given by the SEA. And also the best achievement was not achieved by the average value. For the second objective of greenhouse gas emissions of low quantity manufacturing process, the highest greenhouse gas emissions were recorded for the electrical consumption at the stages of withering and drying section as those were recorded as the highest consumption stages. According to that, the total emission due to the electricity use were recorded as 0.5 for the electricity consumption as an average for the all three factories and also here in the factory 2 was recorded as the lowest greenhouse gas emission. When we compare with the previous studies with our results, the highest energy consumption was recorded within section. It also same as the previous studies. When we compare the withering and drying stages, most of the scientists have noticed that the withering and drying at the current instant bears excessive significance in black tea manufacturing for the quality of the final product and also especially for the reduction of greenhouse gas emissions which happening in the withering and drying stages. And also, most of the scientists have noticed and reported that at the stage of consumption, it means that the consumer level has been reported as the highest greenhouse gas emission stage from the whole life cycle stage. And also when we consider about the cost of production, it can be reduced if we consider more about the withering and drying stages for reduction of energy. For the third objective of greenhouse gas emissions in use of different energy sources, in here we are especially concerned about the firewood use, diesel use, and also electricity consumption. From those three, the highest greenhouse gas emission was recorded from the firewood use. And also the total emission was recorded as 5.0 as an average for all three factories. Finally, for the total greenhouse gas emissions in the low country orthodox black tea production, we were able to calculate the average value as 5.08 with a standard deviation of 2.0 for the low country orthodox black tea production. So when we consider the previous studies with our calculations, the basic greenhouse gas emission value for the black tea production in Sri Lanka for the whole life cycle is around 32 kilogram of carbon equivalent for making 1 kilogram of made tea. So considering that value, 15% impact is due to the manufacturing of black tea in Sri Lanka. It means there's a 15% impact due to the manufacturing process of black tea in low country of Sri Lanka. When we consider our values with the other countries, we are higher than the Taiwan condition. And also, when we consider about the Chinese condition, their tea processing costs for the 41% of impact in greenhouse gas emissions when considering about the total production. When concluding the session, the highest specific energy consumption was recorded from factory 1, and also the average was recorded as 0.8 kilowatt hours per 1 kilogram of made tea for low country tea factories in Sri Lanka. The highest emission has been reported at the withering stage, and also total average emission for the gate-to-gate orthodox black tea production process is around 0.5 kilogram of carbon equivalent for kilogram of made tea in low country Sri Lanka. And also the highest emissions were identified at the withering and drying stages, and those were ranged around 26% to 35% from the total emission of orthodox black tea production. And further, in concluding, the highest emission has been reported from the firewood as when considering about the energy sources. Consequently, the total gate-to-gate carbon footprint of orthodox black tea manufacturing process in low country Sri Lanka is 5.08, with having a standard deviation of 2 for 1 kilogram of made tea in Sri Lanka. These are the references of our study, and thank you all.
Thank you, Ms. Pereira. Now the presentation is open for discussion. Any questions from the audience?
Thank you, Ms. Nimeshika, for your interesting presentation. This is just actually clarification. I saw in the presentation when presenting results, there are like results have been presented up to like 4 decimals. Is that significant, Meshika?
Yes, madam. Because that we have taken that the functional unit for the 1 kilogram. So because of that, the carbon footprint value is very less. But in considering the Sri Lankan situation, we are not producing as millions per 1 month. Because of that, we have to take that accounts. It means that the production in kilograms, it means just a small amount when we compare with the other countries. Because of that, we take that the functional unit as 1 kilogram of made tea. So according to that, we have to keep that the 4 decimal points for making kind of significance.
So the
yes, ma'am. Yes.
Okay, thank
yes,
you.
of course.
Okay, thank you, Nimeshika, for the nice presentation. Just for my understanding, because we are dealing with agricultural technology, your sample number was 3.
Yes, of course.
And you measure some indices like electrical consumption, and some of the area they are not using electrical consumption for the curing and drying process, they use firewood.
Yes, of course, sir. But at the start of the motors, they have to use electricity. That's why we have included under electricity consumption also. For the starting of that motors, they are using electricity.
Okay, then in some, I think all the 3 replicate that you use may have same conditions. That mean area, the kilograms, and the extent.
Actually, sir, these are not 3 replicates. So we selected purposely these 3 factories as the highest production factories for low country Sri Lanka. So already from the Sustainable Energy Authority, they have given the benchmarks for specific energy consumption, but they do not concern about the stepwise process. But when we consider about the factory level, actually they require a kind of a study to assess the impact of their stages for the electricity bill and the carbon footprint. Because of that, we fix some kind of energy meters for that machines, and we analyze the energy data there. So that was the reason. Actually, these 3 were not replicates. So
they
just
are separate.
yeah, they are separate.
Yes, actually,
but
that's what we need some benchmark to assess those things.
Yeah.
In that sense, the research is ideal and timely important. So what I'm telling, basically, we look into the parameter which we generate the carbon dioxide like gases. Better to find out there any source of carbon emission which is not being actually significant in the system, like electricity burning, like boilers. Those are we targeted. So what we want to know whether the carbon dioxide can be emitted even during drying processes, curing processes, and like other things. No, that may be minute and not significant. Even that you can include those parameters also, I think that may be valuable, because when you go to up country, mid country, that area where most of the people use firewood rather than using electrical dryers or boilers or like things to get the steam and dry. That's what I'm just giving you some idea. Please look into the parameter which is not significant, but that has some contribution.
yes,
Okay,
of
thank
course.
you very much.
So we have in here considered about the firewood also. So for that, for calculations, we have considered about the conversion factor of the firewood for that. So it is very low from the other sections. Actually, it's not significant.
Okay,
That's
thank
the
you very
reason.
much. Okay.
Thank you, sir.
So, Ms. Pereira, I have one question. I think one of the key findings of your study is that the highest carbon footprint is given when firewood is used as the energy source compared to diesel, right? So, if we consider the current fuel crisis in the country, do you expect different outcomes from your study?
Yes, of course, because as a suggestion also we would like to give that kind of a thing for the factories, because most of the time they are using, normally they are using the current at the daytime. So because when they are taking their electricity bills, there are 3 phases called as day, peak and off peak. So basically they are doing all the activities in the daytime. If they can do their most of the activities in the off peak, then they can reduce actually their electricity bills, because that the payment is less in the off peak time than the daytime. So beyond that, especially as the technological side, we can use kind of variable speed drives for the withering motors and also high efficiency motors for the dryers. So those are the technical inputs that we can apply for the process in means for the reduction of electrical consumption and also the reduction of carbon footprint.
Okay, thank you very much, Ms. Pereira, for your informative presentation and answering the questions. So, any other questions from the audience? Okay, so I think we can move on to the last presentation of the technical session number 2. The title is The Quality of Wet Type Cat Food Available on the Sri Lankan Local Market, a Nitrite Analysis and Escherichia coli Toxicity Analysis. This study is authored by S.K. Suryarachchi, J.J. Vevalwala, Vajrapani De Silva, and Samira R. Gunatilaka. The paper is going to present by Ms. S.K. Suryarachchi.
Good day, everyone. I'm Kavya Suryarachchi, and I'm here to present my research work on evaluating the quality of wet type cat food available on the Sri Lankan local market through a nitrite and an E. coli toxicity analysis. So this research work was carried out as a collaborative project between the Department of Agricultural Technology, University of Colombo, College of Chemical Sciences, Institute of Chemistry, Tech Research Institute, Sri Lanka. So if we were to first understand why this research is important, cats have now become the second largest pet food consumers around the world, second only to dogs. And out of all those, pet type cat food has low shelf life because of them containing raw animal products, ingredients, and therefore they require additives and preservatives to extend life. Sodium nitrite, a multifunctional additive, is the most commonly related additive that is being used for this purpose. But we should know that if we exceed these addition limits of 20 milligrams of sodium nitrite per 1 kilogram of sample, as according to the FCO, they can cause numerous ill effects such as methemoglobinemia, cancer, and diarrhea in cats. Moreover, E. coli that is being analyzed in the study is also of importance. Since wet type cat food contain high raw meat diets, the raw meats can facilitate the growth of the Escherichia coli, a bacterium commonly associated with meat-based products and is known to create toxicities in the host. Therefore, the research question and the hypothesis is based around the fact that if the manufacturers of these wet type cat food products follows the correct protocols and regulations and prevents the accumulation of nitrite and E. coli. Therefore, based on that, we formed the research objectives of first to identify and quantify the nitrite levels found in the cat food samples and to compare these results with the regulatory set limits. Then to identify the presence of E. coli bacteria in these samples, and through these to state the quality of the store-bought wet type cat food samples. So the analysis part of the study was twofold: a nitrite analysis, the chemical component, and an E. coli analysis, the genetic part. 4 brands of wet type cat food commercially available on the local pet food market were chosen as a sample for the analysis. And we got to know that the product samples were manufactured and distributed from one place for each brand separately, and therefore the locations of buying were disregarded, and only the date of manufacture and expiry was considered to give the uniformity to the samples that is being analyzed. The properties of the chosen brand samples are given here as on the table. First, when we consider the chemical analysis part of the research, the grease assay protocol was chosen as the method of analysis because it is a simple, rapid, commercially cost-effective method, giving both qualitative and quantitative results, having excellent LOD values. The grease assay is basically where the sulfanilamide, one of the 2 grease reagents, reacts with nitrite that is there in the sample in acidic media to produce a diazonium salt, which then couples with the coupling reagent NED to produce an azo dye, which is in purple in color, which we detect through UV spectroscopy detection at 450 nanometers. So the sample pre-treatment was carried out by homogenizing and precipitating proteins out, and finally to obtain an acidified final extract. The main part of the chemical methodology was first to validate this method, which was done by carrying out evaluating detection limits, obtaining spike recoveries, and matrix effect values. So first, the standard curve, this was done through, first the standard curve was obtained using distilled water, and then the matrix match plot was obtained using samples extracted, and these 2 plots were compared to obtain the matrix effect, through which we determine whether to use a standard addition curve or an external standard addition curve to find the nitrite levels of the unknown samples. So then the unknown samples were pre-treated, and the detection and the quantifications were carried out according to standard addition methods, and the unknown concentrations were obtained. So when moving on to the E. coli analysis part, it was carried out by first the sample pre-treatment, which was then stored at -20 degrees till later use. Then the LB agar culture media was prepared and poured into petri dishes and was allowed to settle and cool. And after this, equal amounts of the before set pre-treated samples were cultured in these separately prepared petri dishes and was allowed for the bacteria to grow by incubating for 24 hours. Once these bacteria were grown, the bacterial sample colonies were identified through the naked eye and separated carefully, and the bacterial DNA was extracted using the boiling method. So 2 DNA detection identifications were done to first confirm whether the grown colonies are actually of bacterial origin using unibac detection, and if yes, were they of E. coli strains using E. coli detection. These were done by using PCR amplifications with relevant related primers, which was then run on an agarose gel with 100 BP ladders and later visualized to obtain results. So these are the PCR amplification protocols, thermal cycles, and primers we use separately for universal bacterial detection and E. coli bacterial detection. The method validation results are such that LOD and LOQ values for the method were 7.7 ppb and 23.3 ppb respectively. The spike recovery values were such that the low spike we obtained a recovery of 29.02, which is low in accuracy, but had high precision as the standard deviation is less than 10%. And the high spike recovery we obtained was 115.61, which was both accurate and precise as the standard deviation was below 10% and the recovery value was between 90% to 120%. So all the 4 brands show significant matrix effects greater than 12% and therefore had to follow a standard addition protocol for us to determine the unknown nitrite contents in the unknown samples. The nitrate contents of the sample of all the 4 brands obtained are given here as follows, and the values range from 4 ppb to 7 ppb levels. Therefore, all the brands did not exceed the regulatory limits set by the FOSMedia, showing a significant difference between the two. No any statistical difference was shown between the said 2 values. And also, no any, if we consider the countries of origin, sample of Thailand-produced brand A had the highest nitrite content, and a sample of Thailand-produced brand B had the lowest nitrite content. Overall, when we consider the average, brand C Sri Lankan produced samples had the highest nitrite content, while a Thailand origin brand B had the lowest nitrite content. Even though some brands have labeled the addition of sodium nitrite, they did not exceed the permissible limits. When we consider the results for the E. coli analysis part, all the 4 brands had shown colony formation in the LB agar media, suggesting of bacterial growths. All the 4 brands contained morphologically similar colonies, which was taken as colony X, and only brand A had another morphologically different colony, which was taken as colony Y for the analysis. Then these obtained colonies were first checked for the bacterial origin, and both the colonies shown a strong band at 371 VP in level with the positive control. Therefore, we can say these colonies were of bacterial origin, and since the colony X and Y represent all the 4 brands, we can say that all the samples A, B, C, and D contain bacteria in them. Once it was confirmed that these were of bacterial origin, the confirmation for the E. coli strain was carried out, and the gels obtained were as given here. Here also both the colonies X and Y gave strong bands at 101 BP mark of the ladder in line with the positive control, and therefore we can say both the colonies contain E. coli strain in them as well, and since X and Y represent all the 4 brands, so we can say all the brand samples had E. coli present in them. However, it is recommended to carry out further studies to confirm the level of positive observation for the presence of E. coli that we obtain through methods such as DNA sequencing. Here we observe a slight band in the negative control, and this can be due to the fact that containing contaminations during extractions. In conclusion of the study, therefore, we can say through the detection limit values we obtain, it suggests that the method is sensitive, accurate, and reproducible, and the recovery values suggest that the method is reliable. And all the samples did not exceed the nitrite permissible levels set by the FOSMedia, values ranging from concentrations of 4 ppb to 7 ppb. And the samples showed positive results for universal bacteria and E. coli, but future studies are recommended to find the level of confirmation of the positive result. And therefore, in hindsight, we can say the samples are in good quality, in good consumable quality when considering the nitrite level aspect, but should be attentive from the microbial growth perspective. So finally, I would like to acknowledge the Faculty of Technology, University of Colombo, Institute of Chemistry, Ceylon, and Gene Tech Research Institute for providing facilities to make this research a success. Thank you, and I'm open to any questions.
Thank you, Ms. Suryarachi. Now the presentation is open for discussion. Any questions from the audience?
Thank you very much for your presentation and submitting your abstract to our ARS. Just to clarify, now I think you have used filtrate for the analysis of nitrate. Am I correct?
Yes.
Yes, then that means you use filtrate to analyze the nitrate. Did you use any technology to see whether there is any sort of retention of nitrate at the solid sample that you have separated from the aqueous phase?
Actually, yeah, we were analyzing for nitrites, and nitrites are water soluble. So, since we are obtaining the filtrate, it can't be residue. It can't be stuck on the residues, and it has to be there in the filtrate.
That means we assume that all the nitrate will come to the aqueous phase rather than stagnated on the solid phase.
Yes.
Yes, okay. Thank you. I have another question. When you go to the gel, second gel, what are the type of primer that you use for amplification of E. coli?
For the second gel, actually, it was the E. coli primers we used.
That means specific primer for E. coli?
Yes.
Yes. In the well, explain what is the X mean?
X means we after culturing, we got two colonies formed, X and Y. So colony
yes.
X represent.
Yeah, with that X with the specific primer, we can see two bands. One is maybe specific, other one is. What can be the reason for having two bands? Because when once we use the specific band, so we should get the entire one band there as a specific. But that may
it can be due to primer annealing non-specifically. It can be due to that. Because of that, we are getting at 250 BP.
Yeah, the 250 BP is the expected band size, but you are.
No. 371 BP is the expected one. No, 101 is the expected one, and 251 is the additional one. So we obtained that maybe because of the primer annealing present during the extraction procedure.
That means that is a false positive band. That means it may be contamination. Oh, you are thinking because there should be a DNA that may be annealed with that specific region as well, because that's what you are getting two band. According to your conclusion, it may be contamination with another bacterium, or is due to false positive binding, or that means maybe due to the annealing temperature.
Yes.
Because if you are getting annealing temperature diverse, that you should get that same band in other lens also. That just we had to you have to check whether that may be due to contamination, no, primer annealing. Okay, thank you very much.
So, Ms. Suryaraji, I have one question. So, I think your study suggests that that these PET feed samples you selected, the sodium nitrite levels are within safe limits, but there is a potential risk for E. coli toxicity. So I would like to know that the major reason for using this sodium nitrite as a food additive is its antimicrobial property, right? So didn't you observe any relationship between the sodium nitrite levels and the level of E. coli you observed?
Actually, no, up to that level, I couldn't bring my study as an undergraduate level because to understand that level, we have to quantify the amount of E. coli present in the samples. So up to that level, we couldn't proceed with our research.
Yes, I think I feel like to know the presence of E. coli important, but I think to know the levels of the presence is also very important, I guess. Yes, thank you, Ms. Suryarachi. Any other questions from the audience?
Yeah, thank you for your nice presentation. So I have actually one question. Did you use any positive and negative controls in the experiment?
Yes, we did.
What
Positive
are
and negative.
those? Yeah. What are those?
For the negative control, without the without sample, we used the PCR pre-nucleus water and everything for the negative control with the master mix, and for the positive control.
Yeah, what I'm asking is the foods which you know that that food doesn't have sodium nitrite, something like that. Did you use such kind of non-positive negative controls in the experiment?
You mean for the genetic analysis part or the nitrate analysis?
Nitrate analysis. Even
yeah.
for that is applicable, yes.
Yeah, for that, we actually use the spike recovery values. So we actually spike the samples initially before, like a known amount. Yeah, we spiked and obtained the results, and
spiking spiking. Did you use for the standard or did you use for the food sample itself?
We used for the food sample itself.
Food sample itself.
Yeah.
Then that's okay. Yes. Thank you.
Yeah.
Okay, thank you, Ms. Suryarachi, for your informative presentation and answering the questions. So I think this is the end of technical session number 2. I would like to thank all the presenters and the authors for their valuable findings and presentations. Thank you very much.
Thank you very much, Ms. Jayaprada. That's the end of the technical session 2. Let's move to the technical session 3. I would like to invite Dr. Rohan Samarasinghe, the head of the Department of Information and Communication Technology, to chair the technical session 3. Over to you, Dr. Samarasinghe.
Thank you very much, Dr. Hansika. So let's start the session 3 sessions. First, I would like to invite the first presenter. The topic is a low-cost optical design for the surface plasma resistance, copper graphene ITO substrate. Authors are L.M. Samaratunga, M.W.I. Chanilka, M.D.R. Devamitra, J.L.K. Jayasinghe, K.D.R.N. Kalubovila, M.S. Gunawardana, H.H.E. Jaiviran, J.K.D.S. Jayanetti.
I'm Lasin Samaratunga. Today, I'm going to present about a low-cost optical design for a surface plasma resonance sensor using a copper graphene ITO substrate. First, I would like to explain the surface plasma resonance effect using a commonly used setup configuration known as the fixed angle Frenchman configuration. As shown in figure 1, the setup contains 5 main components: a laser, optical detector, prism, sensor chip, and analyte, which is illustrated inside the flow channel. The sensor chip contains a metal layer and a dielectric layer from bottom to top. When the polarized laser is incident on the metal glass interface, a standing electromagnetic wave will be generated, which is known as the evanescent wave. The evanescent wave will oscillate the free electrons in the metal layer, inducing a second electromagnetic wave, which is known as the surface plasma wave. At a certain angle, which is above the critical angle, the surface plasma wave will oscillate with a maximum amplitude, generating surface plasma resonance. Hence, upon the resonance angle, there will be a significant reduction of the reflected laser beam. In a graph of reflection versus incident angle, there will be an intensity dip at resonance. The resonant angle is highly dependent on the reflective index values of the layers. Next, I would like to discuss about advantages and drawbacks of SPR sensors. SPR sensors are mostly used as molecular detectors such as protein detectors, glucose detectors, and salinity detectors. The main reason for use of SPR sensors as molecular detectors are the high limit of detection, the high sensitivity, and higher signal-to-noise ratio values. But SPR sensors also have some disadvantages, such as use of rare metals, need of index matching materials, need of specific lasers. All the above mentioned facts will increase the cost of SPR sensors significantly. Therefore, it is vital to produce new designs for low-cost SPR sensors. In order to reduce previously mentioned disadvantages of conventional SPR sensors, we suppose that by using copper as the metal substrate, the cost will be reduced significantly. Also, it is suitable to implement a graphene layer to protect the copper from corrosion. Due to the broad plasmonic range of copper, a 650 nanometer laser would be effective since it is a commonly available laser. And finally, instead of a glass prism, a water prism made out of a transparent case will be much cost effective. A hole will be cut out of the prism case where the thin film is connecting in order to directly contact the water to the thin film. Next, I would like to explain the methodology carried out to determine the optimized copper layer thickness and the graphene layer thickness of the proposed SPR sensor. First, the 4 layers were identified for the sensor chip from top to bottom: graphene, copper, ITO, and glass. The thickness of the ITO and glass were determined from laboratory produced ITO glasses. As for the next step, free version software known as WinSpal was used to generate reflective intensity versus angle graphs by solving Maxwell equation and Fresnel equations for a given SPR event. As for the third step, in order to determine the optimum copper layer thickness without the graphene layer, all the initial conditions were specified as follows. The laser was determined as 650 nanometers depolarized light. The prism shape was defined as semicircular. 2 independent variables were defined, which were incident angle and the copper layer thickness. ITO and glass thickness were specified, and finally the reflective index values and the absorption values of each layer were specified relative to the laser wavelength. From the simulation, the optimum copper layer thickness was obtained. Finally, separate simulation was initiated to determine the optimum graphene layer thickness with the use of the obtained copper layer thickness. The result and analysis for the copper layer simulations are as follows. In figure 3, it is possible to observe every copper layer exhibits SPR dip around 52 degrees to 55 degrees, and additionally, distortions can be observed due to the water glass layer reflective index mismatch. From the intensity versus angle graph, the SPR dip positions for each layer were identified and mapped separately in figure 4, which clearly indicates a minimum intensity value for 40 nanometers of copper layer thickness. Similar to copper layer simulation results, the graphene layer simulations also has distortions in figure 5, and the SPR effect has occurred around 53 degrees to 55 degrees for all graphene layers. The slight resonance angle shift is due to the reflective index change in the sensor chip from the newly introduced graphene. Unlike the copper layers, it is possible to observe in figure 6 that graphene layer thickness increment will reduce the SPR dip intensity. Finally, conclusions can be made that optimum copper layer thickness for the SPR sensor would be 40 nanometers. Minimum graphene layer thickness of at most 1 nanometer would be suitable for the SPR sensor. Distortions can be reduced by increasing the reflective index of the prism, such as using glycerine instead of water at the prism material. Further developments can be implemented by experimentally constructing the SPR setup and obtaining data. These are the acknowledgements I wish to share. And finally, thank you for listening.
Okay, thank you very much for the presentation, and we will open this session for the question and answers. Those who are joining via the Zoom also can ask the questions from the presenter, Mr. Samardu. Okay, there is one question. Several disadvantages. One is use of rare metal accuracy. How did you overcome not only this? There are 3 or 4 disadvantages. How did you overcome such disadvantages using your model?
Hello, sir. Can you hear me?
Hello. Can you hear me?
Yes, sir. I can hear you.
Yes, then you have mentioned several disadvantages of your model. So how did you overcome such disadvantages? Have you had a special adjustments or method for this one?
So, sir, the disadvantages are mentioned for conventional SPR setups. So in my proposed setup, I have overcome those disadvantages by if there is a disadvantage by using rare metals, I have used copper instead of like gold, silver. So that means I can reduce my cost in my setup instead of using highly expensive metals. And in index matching materials, so in my setup I have used water instead of glass. So since I have used a liquid material, I can directly contact the thin film without using another index matching material. And for the final disadvantage in conventional SPR setup, I have mentioned that if you use metals such as gold, like gold and silver, yes, so we need to use specific laser wavelengths to excite those free electrons in gold, silver. So in copper, there is a broad plasmonic response range, which means we can use a set of wavelengths instead of a specific wavelength. So that's why I have used 650 nanometer laser, because it is the most commonly available laser. So copper will excite at 650 nanometers, and I can reduce the cost by using a most commonly available laser, sir.
Okay, thank you very much. Are there any questions?
Thank you, sir.
So, thank you very much for the presentation then. Let's move to the second one. Raman spectroscopy analysis for Bovine serum albumin using electro-deposited copper on indium tin oxide as a SERS substrate. Authors are SPNN Senadira, UD Rodrigo, LM Samartunga, JLK Jayasinghe, KDRN Kalvovila, NI Abesinghe, UD Kotarachi, JVP Fernando, and JKDS Jayanthi.
Good afternoon, everyone, and welcome to my presentation. I am Nimesha Senadira from Department of Medical Laboratory Sciences, KDU. The topic of my presentation is Raman spectroscopy analysis of bovine serum albumin using electrodeposited copper on indium tin oxide as a surface-enhanced Raman spectroscopic substrate. First, let me lay out the contents that I'm going to talk today. First, I'll start with an introduction and move on to the importance of detection of proteins and different methods of detection, the use of Raman spectroscopy and surface-enhanced Raman spectroscopy for protein detection and analysis, the methodology and the results. So let me begin the introduction by explaining what is bovine serum albumin. Serum albumins are the most abundant proteins in the circulatory system of wide variety of organisms. Bovine serum albumin, also known as BSA, is a protein derived from bovine blood plasma. It's a small, stable, and moderately non-reactive protein and therefore often used as a protein standard in laboratory experiments, such as Bradford test, bromocrysyl green test for albumin determination. Equally, the analysis of it is compatible with wide range of chemicals and reagents. And most importantly, as the structure of BSA is very similar to human serum albumin, it has been widely used as a model protein in research instead of human serum albumin. Why detection of proteins is important? Well, do you know there are 100,000 different proteins in our body and they perform different functions such as transporters of exogenous and endogenous substances, transporters of ions, act as building blocks, and sometimes as pathogens in biology. For biological studies and clinical diagnosis, detection and determination of proteins is very important. For biochemistry, pathology, and the clinical diagnosis, it is necessary to quantify the amounts of proteins in living systems. For all these, we require highly sensitive and selective detection methods. Let's now take a look at the different protein detection methods. The method of protein detection should be accurate, fast, sensitive, and reproducible. Currently, spectroscopic techniques, enzyme-linked immunosorbent assays, Western blot, and electrophoresis methods are used for the detection of proteins. All these approaches have reasonable selectivity and sensitivity, but still there are some disadvantages such as complexity of the procedure, poorly reproducible, and time-consuming. Therefore, the detection of minimal amounts of proteins in the shortest time using a biosensing tool with simple design and high precision is very desirable. This leads directly to my next point, which is the use of Raman spectroscopy for the detection of proteins. Raman spectroscopy is a scattering phenomenon. When an incident radiation strikes a molecule, elastic and inelastic scattering of photons will occur. If the incident photon energy is higher than the scattered photon energy, we call that Stokes-Raman scattering. By measuring this energy difference, information about the vibrational energy and the frequencies can be obtained. These energies are molecule specific and provides information about molecule structure. Especially for proteins, this provides useful information about certain regions. For example, drug binding target sites. It provides insights on functional moieties, amino acid residues, and secondary structural changes of proteins. It is highly sensitive and has a short measuring time. It requires low sample volume, none or less invasive, and non-destructive. To enhance the weak Raman signals and to get more resolved spectra, we can use surface-enhanced Raman spectroscopy. The Raman effect is intrinsically weak and not sufficient to detect low concentration of the analyte. SIRS uses metal nanostructures typically made of gold or silver, and currently copper-based SIRS substrates are also being used as alternatives. Laser excitation of these metal nanostructures resonantly drives the surface charges, creating a highly localized plasmonic light field. When a molecule is absorbed or lies close to the enhanced field at the surface, a large enhancement in the Raman signal can be observed. Let's now take a look at the Raman spectra of BSA that I obtained using the hanging drop method. According to the literature, we can see the overall pattern of the spectrum is same, and the prominent peaks like phenylalanine peak at 1002, CH2 stretching band at 1450, and amide 1 band at 1650. To enhance the weak Raman signals and to get more resolved spectra, next I use SIRS using copper-based nanostructures. Copper is not traditionally used in SIRS, but it does have 10 to the power 5, 10 to the power 7 factor of enhancement, and it's inexpensive. Also, according to the literature, albumins are known to form bonds with divalent cations like cobalt, copper, nickel, and among them, copper has greater affinity towards certain binding sites. Considering all these, in here I have used copper deposited indium tin oxide thin films as the SIRS substrate. Now I'll brief the methodology. First, copper thin films were electrochemically deposited on indium tin oxide glass substrates. Then, 10 microliter of BSA standard solution drop was placed on copper-coated IPO thin films and Raman spectra were acquired. Finally, the data were analyzed and peaks were identified using existing literature. According to the literature, I identified the prominent peaks and displayed them on a table. As you can see in the spectra, now the spectrum is more resolved and it seems like there is an enhancement of the prominent peaks of PSA. But still, we have to optimize and get in the process to increase the enhancement factor. In conclusion, these findings support the feasibility of analyzing BSA standard with Raman spectroscopy as well as the application of copper-based SIRS materials to it. And this is from the preliminary work I have done using copper-based SIRS materials, and in the future, further studies will be carried out applying copper-based SIRS materials, and the system will be optimized accordingly. These are the acknowledgements I want to mention. Thank you for listening.
Thank you very much for the presentation. And now we are going to open this presentation for the question and answers. Those who wish to ask the question from the presenter, I will invite them. I have one question. You have mentioned that the existing method for Boolean BSA detection, and you introduced the method using the Raman spectroscopy analysis using the copper deposit electro deposited copper on indium tin oxide as SCRS substrate. So the previously you mentioned the existing method, actually you have categorized those methods for the accuracy and sensitivity, using the accuracy and sensitivity. Then your proposed method, which you are going to maintain the accuracy or sensitivity or both?
So actually what I wanted to mention was like if we want to identify or detect a protein, the protein detection method has to be accurate, sensitive, fast and reproducible.
Actually, it's not a categorization. I just wanted to mention that the protein detection method has to have these kind of properties. So the existing methods does have this up to reasonable level, but when we compare with the Raman spectroscopy, like it has like advantages of like it is highly sensitive, and it can provide useful information about the smaller regions like up to bond level, and it has a very short measuring time, and the like easy to like the sample preparation steps are very simple and it is easy, and it requires very low amount of sample volume, and the sample gets like none or less invasive and non-destructive. So that's why I wanted to introduce the Raman spectroscopy for the detection of proteins.
So is that your solution low cost or considerably higher than the existing one?
It is low cost, sir. Due to the low sample volume and short measuring time, it takes low time, and the like the reagents, the use of reagents and everything, it is very less. So the cost will be like lesser than the conventional methods, and the sensitivity also higher.
Okay, thank you very much for the presentation, and you have done a valuable presentation. Let's move to the final presentation. Instances plant growth dynamics reveals ozone sensitive intrinsic and nanometric fluctuation in growth rate. Authors are B.L. Sanjay Tilakaratna, Uma Maheshwari, Raja Gopalan, and Hirofumi Kadono.
Good afternoon, Chairman, Chief Guest, the Vice Chancellor, University of Colombo, the Dean, Faculty of Technology, University of Colombo, keynote speakers, professors, lecturers, and my dear colleagues. Today, I would like to explain a novel optical sensing method that can be used to measure the growth dynamic of plants in different environmental conditions. The topic of my today's presentation is instantaneous plant growth dynamics reveals ozone-sensitive intrinsic nanometric fluctuation in the growth rate. There are three main collaborators for this project. I am Sanjay Tilakaratna, representing Faculty of Technology, University of Colombo, Professor Uma Maheshwari from Riken Brain Science, Saitama, Japan, Professor Hirofumi Kadono, representing Saitama University, Japan. Moving to the presentation, some of you may be aware that a tropospheric ozone or ground level ozone is a secondary air pollutant produced by the photochemical reactions of nitrogen oxide, carbon monoxide, and volatile organic compounds. This ozone is a very harmful air pollutant for the plants. Ozone react with the primary to the plants through the stomata and immediately interact with the water and other cellular compounds and make this type of reactive oxygen species. Superoxide radicals, hydrogen radicals, and hydrogen peroxide. These reactive oxygen species will damage various cellular components in the plant leaf. As we can see this type of cellular level changes as well as microscopic level changes in the plant. So in this figure, you can see how ozone affect the plant growth. There are three chambers, charcoal filtered, ambient and ambient 1.5 ozone. We kept the plant in these three chambers and observe the variation of the growth. So we can see there's significant reduction of the plant growth as a result of the ozone. So this figure shows the same condition applied for the, and you can see how plant leaf growth varies because of the ozone effect. There's a significant reduction in the plant growth of the leaf because of the ozone. Now you can understand why we need to study about this ozone effect against the plants. Okay, so still we are using conventional measuring techniques to measure those changes in the plant. Biomass change, height change, counting, chemical analysis, almost all the methods are up to micrometer scale. Sometimes we have to wait to take the harvest. That means it will take long period of time to take the measurements. So that is why we need highly sensitive techniques that can quantify the growth dynamic of plants in short period more precisely and observe the immediate response of the plant with ecobiological factors. So those sensitivity limitations can be achieved using the optical interference method. So those optical interference method guaranteed very high sensitivity with high reliability. And we can do the real-time and non-distracting measurements using the optical interference methods. Available optical interference methods are very limited because research application in the experimental environment, because sometimes the instability of the living object and speckle decoration due to the continuous changes in microstructure and multiple scattering due to the light penetration. However, we were able to overcome those sensitive limitations and develop some novel method called statistical interferometry technique. This technique is capable to do a real-time monitoring in sub-second in the sub-nanometer precision. So we can have following advantage about the SIT. So we can, as I mentioned earlier, we can do sub-nanometer precision in per second time period. We can do the real-time monitoring with the very short period of time. It is a very simple optical system and with high accuracy, and we can do measurement in non-contact with the object, and we can apply the scattering object like plants. So those are main advantages behind this SIT. Now I will explain the principle of the SIT. However, I skip the detailed principle of the SIT because of the time limit. So briefing about the SIT, some of you may be aware that when rough surface is illuminated by a laser, a fully speckled pattern, speckled pattern appeared in the diffraction field as a result of complete random wavefront like this. So this speckled pattern is, we utilize this speckled pattern as the primary object of the growth rate measurements. So now what you can see here is the experimental setup of the SIT. The leaf of the plant kept in front of the two parallel laser beams. A laser beam is split in the two beams by means of this prism P1, and these two beams are made parallel by means of this prism P2. The piezoelectric transducer is given a phase modulation between the two laser beams. During the growth of the leaf, the random wavefront interference patterns change, and corresponding those intensity distribution are successfully acquired by the CCD camera and stored in the frame memory. When there is a delta X expansion between the two illumination points, the change in the phase between the two interference speckle field is given by this equation. So now you can measure the growth of the leaf using this equation. So this is how we clamp the leaf for the SIT measurement. This is rice leaf. We kept the there's a clamp, and we clamp the rice leaf like this. And this is the real system of the ozone measurement for the short-term ozone measurement for the SIT. This is ozone chamber. We kept the rice plant or other plant inside this chamber, and we applied the ozone and charcoal filter air to the chamber and measure the growth behavior. This is the optical setup of the SIT. So I will show you which data taken from the SIT. So and I will explain how we are taking data measurements using the SIT and how to validate this data. So figure A shows the growth of the leaf for 1 hour time period. You can see the measurements were in the micrometer range. Figure B, the growth of the leaf was calculated for the 1 minute time period. The y-axis is still in the sub-nanometer scale. Figure C shows the growth of the leaf for 5.5 seconds, a very short period of time. Now we can have a very powerful tool to monitor the growth behavior of plant in ultra short-term time period. I think so far in biology no one studied or experienced such a plant behavior. The slope of the fitting line was calculated as the growth rate by normalizing the beam distance. So when we plot this growth rate over 1 hour time period, we can see this type of fluctuation. So we name this fluctuation nanometric intensity fluctuation. Now you can see this fluctuation in different stages of the plant: young leaf, mature leaf, and dead leaf. So we can see this fluctuation all stages of the plant in different scales. So young leaves show the high standard deviation values, mature leaves shows the some small value, and very small value was shown by the dead leaf. So now you can understand this NIF is that this NIF is differ from the conventional growth rate measurements. So it is the fluctuation observed in all part of the leaf, but it is now we can say it is the intrinsic property of the plant leaf. So they differ from the conventional measurements, and it's the intrinsic property of the plant leaf. So now I will show you experiment data which we gathered from the SIT. So this is the short-term ozone exposure that we, as I showed in the image, we applied ozone and charcoal filtered air to the chamber and measure the growth rate behavior. So we had control sample and experimental sample. We applied this whole time period for the experiment 7 hours. We divide in 3 time interval: 1 hour charcoal filter, 3 hours ozone, 3 hours ozone to charcoal filter, and again charcoal filter in 3 hours. So altogether we have 7 hours. So a control experiment for 7 hours we applied charcoal filter there. So you can see how this standard deviation variation. Though almost the standard deviation is almost equal in 3 time stages. Okay, in the control sample. But if you move to the experiment sample, so we applied the charcoal filter there for first hour, and second 3 hours we applied ozone, and another last 3 hours we applied charcoal filter there. You can see how ozone affect for the fluctuation. Okay, so there's reduction in the fluctuation as a result of the ozone exposure. So now you can see we would be able to identify the ozone effect within a very short period of time. Very short period of time, within 3 hours time is enough to identify the ozone effect for the plant. Okay, we under this control the temperature and humidity were controlled and almost constant in both cases. And now I will show you long-term exposure of the plant. This is studies plant. We had 3 chambers there: charcoal filtered, ambient air, and ambient 1.5 ozone chamber. So under this, we kept radish plant and 30 to 40 days. All radish plants were used for this experiment. So you can see charcoal filtered air has high growth rate of the plant leaf, and ambient air we can see slight reduction, and in the ambient 1.5 ozone air there's also significant reduction in the standard variation. So in the fluctuation. So this is for long-term exposure, that means 30 to 40 days all plant. So that we applied for 30 to 40 days ozone in this condition. Okay, so that means there's accumulated ozone in the plant leaves. So this is plant. So we can this is also a long-term exposure experiment. 30 to 40 days all soybean plants were taken, and this is how that ozone varies because of the fluctuation varies because of the ozone effect. So standard deviation slightly significantly changed in 3 conditions. So finally, I would like to conclude my talk with the following points. Statistical interferometry technique offers high sensitivity up to sub-nanometer scale measurement in very short period of sub-second time. We found new phenomena called nanometric intensity fluctuation. The NI were found to be influenced by exposure to ozone and differentiate the rice cultivars and sensitive enough to differentiate the ozone levels. So, and I finally conclude this following point. So, statistical interferometry offers high potential with very high sensitivity and much feasible exploration of new insight of the short period dynamics of the biological object like plants. And thank you very much.
Thank you very much for the presentation, Dr. Sanjay. So this will open for the question and answers. So Dr. Jayani will ask a question from you.
Hello. So Sanjay, it's a very nice presentation. I have one question. So you have measured the elongation of the plant, upper body of the plant, right? So have you studied about the root system of the plants?
Yes, Dr. Jani, yes. Yeah, we are studying now root system of the plant. We are getting same type of result.
Okay, thank you, Sanjay.
Yes, Sanjay, I have another question. You have used the ozone sensitive, but when you consider the environment, we have some carbon emissions and heavy metals also. So how did you, why don't you measure all those things also, the carbon and other things?
Yes, actually, Dr. Rohan, Chairman. So we measured, we are currently measuring those changes in the we apply those stresses to the plant, and we are measuring those changes using this SIT. We are getting good result because for example, heavy metal applications and other polluted gases, if we apply as stresses, so then we got reduction in the fluctuation. The thing is, we are now that our scientists in the other groups advise us to find the reason for SIT. Why is this fluctuation happening? Now we are studying that reason. What is the reason behind this one? It may be some instrumental conductance or instrumental opening or closing, or some other water molecule changes. So we are studying that one. So as you mentioned, we are studying those areas also. We are getting good results. We will publish those things in our in next stages of the research.
Also, you have identified the nanometric fluctuation in growth rate. So how does it influence for the growth of the crop of that plants? Is that?
Yes, as you can see I show you 3 plants: rice plant leaves and soybean and radish plants. So the fluctuations are different plant to plant. Okay, so nanometric fluctuation differ from the plant to plant. So rice plant is below 10, radish plant is around 30, and the soybean around 20 range. So when we are analyzing that, we need to normalize this data. So and the different conditions or different fluctuation we can see, but we are when we are analyzing that, I didn't see the normalized data to you, but when we are analyzing that data, we need to normalize it.
Can we overcome this situation using the greenhouses?
What do you mean by overcome? So we can apply. We can apply to check the plant. Actually, this nanometric intensity fluctuation is the kind of health monitoring parameter of the plants. So we observe that if the health is the plant is not good. For example, dead leaf, you can see that that fluctuation is very low. Okay, so and that if we have some visible injuries in the plant leaf, so with fluctuation is also getting low. So that is why if we have some influence, give some stresses to the plant, the fluctuations are getting low. So we believe that it's the kind of a health monitoring parameter in the plant. So as you know, you may know that intercellular carbon dioxide, so other parameters in the photosynthesis parameters. So those are indicating that indicating the health of the plant. So that we also want to highlight that this fluctuation is also kind of a health indicating health condition of the plant. So we can apply greenhouses also that to check the plants are in good condition or not. Within short period of time, we can monitor that.
Okay, thank you very much. Are there any questions? No, thank you very much for the presentation and answers for the questions. And this will conclude the session 3. And I thank all the presentators to help to make a truthful session. And thank you very much for you all.
Thank you very much, Dr. Samarasinghe. Let's move to the technical session 4. I would like to cordially invite Dr. Chaturika Silva to chair the technical session 4. It is over to you, Dr. Chaturika.
Okay, welcome all of you to the fourth technical session.
A robust
of
multi-factor
the
authentication method for secure
annual fourth technical session of the annual research symposium. So we will move to the first presentation, a robust multi-factor authentication method for securing electronic payment transactions. The authors are N.R. Samrasinghe, N.N. Tilakaratna. So the presenting author is N.R. Samrasinghe.
I am Rohan Samrasinghe from Department of Information and Communication Technology of Faculty of Technology. So all of you may have the credit or debit cards from your banks, and electronic payments or the transaction are getting complicated day by day as people are using these cards to make the payments, and hence electronic transaction security has become a common problem worldwide because of the cyber attacks, loopholes of some authentication methods, as well as weak authentication methods and users are not aware of the importance of the strong password and lack of security alert of the electronic payment method. So unless the electronic payments are not secure due to the unauthorized access to the credit debit cards, the people are reluctant to use the cards for the payments. And financial institute may have several $1 million losses due to this weak authentication methods. But the banks and other financial institute always promoting the online and cardless transactions through online banking and credit debit card payments. And using such methods, the bank can enhance the profit by minimizing the operational costs. So let's see the challenges of the credit debit cards. These credit or debit cards, theft has evolved and developed drastically in recent years. Fraudsters employed skimming tactics as well as card credential steal from the databases, sniffing and packet intercapturing, and shoulder surfing and skimming. They use these methods to get the passwords of these credit or debit cards. So normally you know how to withdraw the money from the ATM machines. You will have to insert the card and enter the password and select the transaction that you want. Then you can get the money. If your password and the card get someone else, they can easily withdraw the money using the card is available as well as the password is there. And for the online payments, use the multi-factor authentication, that is one-time password with the PIN number that can be used for online payment. So, but when you consider the credit debit card, we will not be able to introduce the one-time password because of the time consuming factor, as well as that is not the practically impossible. So we have to have a problem how to protect the credit or debit cards from the vulnerable accesses. So we will how to overcome these challenges. Actually, however, single factor authentication is no longer deemed sufficient for user authentication, and it's regard to insecure for high risk financial transaction involving access to customer information. So then also we can use the multi-factor authentication to safeguard the web transaction. However, that means one-time password, OTP with the card credentials. So we can narrow down this research only for the credit debit card withdrawals as from the ATM machines and payment from the point of sales devices. So we are proposing a novel solution that would allow the user to authenticate every ATM or post transaction via the unique smartphone app by measuring smartphone app or geolocation-based technique to see whether the user, whether to verify the actual user. So we can use the geolocation-based system to authenticate the payment transaction. For our proposal system, we can minimize the cost and waiting time to complete the transaction. So we propose two methods. One is introduce a geolocation-based solution with the REST API, and also we can introduce the mobile app for the card holders to prevent these vulnerable transactions. We'll discuss these two solutions. So if we use geolocation-based solution and the mobile app, first of all, we have to change the software of the system without changing or adding any hardware component, just to do some changes of the software and location, allow the device location access. Actually, we have to register the mobile phone with the card. So what we propose is, if you want to withdraw the money from the debit card, we have to insert the card. When you insert the card, first of all, the machine send an empty packet to the registered mobile phone and get the acknowledgement. This is actually see whether you and your mobile phone is with you or in a certain radius. That is, for an example, 5-meter radius, you and your mobile phone is there. If so, the machine allows to do the transaction. So something like that. The user can make a request, the user can insert the card, the ATM machine through the host computer send a request to the bank computer, and bank computer send the empty packet to your mobile phone and get the geolocation and compare this geolocation and the distance and allow the customer to withdraw the money. Also, next one is, when you want to make payments using the POS point of sales machines, the same scenario can be implemented. That also send a packet to the mobile phone and get the request and see whether you and your mobile phone is in a certain radius, certain meter radius. If so, the card payment is success. Otherwise, it will give the message that the card pay is failure, so that if someone steals your credit or debit card, they cannot make the payment because they don't have the valid mobile phone with them. And also, in the second method, we propose the mobile app. In the mobile app, we can activate and deactivate this authentication, multi-factor authentication, and also we can limit the withdrawal of the purchase limit and hand over the card to the wife or the children, and by deactivating the card, then they can purchase only this limit. And also, if your card is stolen, you can block the card from the mobile app. Also, you can limit, when you travel abroad, you can limit the countries that you can purchase from the credit or debit card. Actually, this app also, when you use this mobile app, the same as the previous method, the server send the message to a mobile app, then the message will pop up and you can accept payment or the withdrawal, then you can only get the money. So, implementation of geolocation-based authentication and REST API, the main objective of this research is to provide the extra layer of security based on geolocation-based technologies. Global position system inputs are utilized with the assumption of the user. The user has an active smartphone location service enabled. And also the mobile application implementation, the transaction authenticate using the mobile app, so that either you can use the geolocation based authentication method or the mobile app implementation to secure your credit and the debit card. When we implement this and when we simulate this, and we could get following results. However, the conditions scenarios such as power off or switch off the location service are addressed to complete the proposed solution, and simulation result shows the high accuracy of the system and could provide the maximum security of the credit and debit card. However, we identify some delays, but it can be negligible. This is the end of presentation, and thank you very much.
Thank you, Dr. Samrasinghe. Now the presentation is open for question. Any question from the audience? Right. Dr. Samrasinghe, I have a few questions. So you have mentioned about two applications, mobile application and the mobile application plus REST API. So there in the mobile application, you specifically mentioned the distance in between mobile application and the ATM machine is 5 meter radius. Why specifically 5 meter radius?
No, you can use any meters, but 5 meter radius. If you want to, you can extend it to the 10 meter radius. That doesn't matter. The only thing is the system checks whether the mobile phone is around you. That's the only thing.
Okay, right. So my next question is, so when you are using the geolocation plus REST API, so you're going to use the GPS sensor based sensing, which is enabled in your phone,
yes.
right? So and the thing is, so most of the POS terminals and the ATM machines are operating indoors. So we know that when it comes to indoors, most of the times the GPS signals are disturbed because of the line of sight with the satellites. So therefore some errors can happen, right? So therefore the results or the expected results may not occur as we want. So how about that?
Actually, we tested all the environment we tested. We did not have such problems. So we came to a prior decision that because most of the ATM machines are open to the door open to the environment, so that we did not
when
have
it comes
such
to the POS terminals?
POS terminals actually there may have some problems of the POS terminals when we want to make some purchases, in-house purchases. So actually there may have some limitations.
Okay. Thank you, Dr. Samrasinghe, and thank you again to that nice presentation as well as for the Q&A answering session. So next we will move to the second presentation, Social Engineering Techniques and Mitigation Approaches. Authors are N.N. Tilakaratne and N.R. Samrasinghe. The presenting author is N.N. Tilakaratne. Over to you.
So good afternoon everybody. So this is regarding the abstract that we have submitted about the social engineering techniques and mitigation approaches. And if I move into the background of the research, so we all know that cybersecurity is an essential component of today's world, because like there are like a lot of cyber crimes are happening and cyber criminals are there, and also the number of cyber attacks are growing and the threat is imminent. And like on the other hand, this cyber attacks, they grow both in terms of their quantity and also like their attack techniques. Technique wise, they have day by day they are using some advanced techniques. So when it's come to this social engineering techniques, attacks, they are a type of cyber attack that does not target the vulnerabilities of the technology. Instead of that, they will actually violate or exploit the human emotions or human psychology. Basically, the attackers will persuade you to do something, and then they will gain information through that. That's how it happens. They basically rely on gaining human trust, as I explained. In general, the human element is the weakest link in the security system of the technology. So the main objective of carrying out this research was like highlighting the social engineering attacks and mitigation approaches towards ensuring better cyber safety, because we have noted there's no much adaptive research has done in this area. So what we have done was we perform a simple review using this keyword search. The information is highlighted already, and this include providing an overview of social engineering attacks, highlighting attack types with countermeasures. So based on the literature, available literature, and from the internet sources, what we have found, there are like 5 main categories of social engineering attack. So like phishing in the sense like attackers basically persuade you to maybe like do something, and then they will gain some information. Like basically they will send some emails to you, like you need to click that information, you need to click this, and you need to subscribe. And uh, unwillingly you will be clicking that link, and then what happen is attackers maybe get your login credential through that. And spear phishing in the sense is same as phishing, but the thing is in spear phishing attack, the attacker first gathers some intelligence about you, whether how is your what is your exact name and your family background and kind of things. They will search through social media, and they will have idea about you, and then based on your preferences they will try to exploit your weaknesses. They might be sending some information, might be sending some information, you need to provide these details or kind of things, and then based on that they will try to gain information from you, some valuable information from you. So that's how the peer phishing and the spear phishing attacks works. And when it's come to baiting, what happen is it's actually they will actually try to violate your greediness and curiosity. That means uh, maybe like they will send some promotion emails, or they will send some message saying that you have won this much amount of prize and you need to provide this kind of information like your credit card number and things, so we can deposit likewise that. So basically they will take you as a bait. So that's what happened in the baiting attack. So when it's come to pre-texting, what happened is the attacker, they will act as some kind of high authority figure, maybe like from police, maybe like some president in the urban council, and they will act as that actual person, and they will try to gain some information from you. That's what happened in the pretexting. So when it's come to tailgating, uh, it's about like the attacker actually tries to gain access to the physical systems. Maybe like if there's a let's say there's an attacker, so he will be initially within the he will be going to a company, and they will then he will basically gain in the trust of their employees, and then based on that he will work as an employee, and then he will try to access into that particular computers, and then through that physical computers he will gain some vulnerable really important information to gain maybe like a competitive advantage kind of thing. So these are the 5 main social engineering attacks that we have found through the literature. And also like in terms of mitigation approaches, we have found that it can be twofold, and because like there are human based and technology based mitigation approaches are there. So that means all the mitigation approaches can be divided into these two. When it's come to human based mitigation approaches, one is policy and auditing. Like if it is organization, you have like regular you have policies implemented, so the employees they need to aware of these policies, and then they must ensure the safety of entire organization through that. If you have ISO 27000 policy in your organization, that means like it explicitly saying to the team employees that you don't need to you don't have to click that whatever the spam emails or kind of thing. So when it's come to otherwise auditing, continuous auditing, it will help you to find out if any anomalies are present in your organization, and if any anomalies are present with this whatever the processes in the organization, and it will help you to identify if there are any attacks as this kind of attacks are present. And like the only other thing is that you can make your employees aware of these kind of attacks, so they will have a better awareness and they will avoid such things. And when it comes to the technology based approaches, like the thing is they are always come at a cost, like physical identity measures like biometrics kind of things, like access control doors kind of things could be there, but the thing is actually they are come at a cost. Then the honeypot devices, it's like a device that you can implement onto your network also, and also like firewalling system. But the thing is, as I told, like when as and also as opposed to this human based approaches, they you need to spend more money on the technology based approaches. So finally the conclusion. So basically human emotions and psychology are exploited in social engineering attacks, and it entails conducting victim research. That means like gathering new information, and like then after gathering new information they will try to win your trust and become friendly with you, and then they will seek vulnerabilities, then they will try to attacks. So it has been a hazard to both individuals and businesses for like all the time. And this is for these kind of attacks, actually there's a no comprehensive security solution, because it don't cooperate with the human trust, and the attacker tries to exploit that human trust and gain some advantage. That's what happened actually. So in terms of the mitigation approaches, human aspect of mitigation can be formed through education, training and awareness, but it has its downside, because you can't always trust your employees in the organization, and you can't even trust anyone. That's the thing. And when it's come technology based mitigation, it is true that it may improve the security, but it has some downsides also, because you need to spend money, you need to spend a higher capital. The thing is, actually these kind of social engineering attacks cannot be removed totally. The only thing that you can do is like like aware the people of such kind of attacks and ask them to avoid from doing such things. That's the only thing that we can do. And also like implementing some policies, like in your, if it is organization, implementing like policies ISO 27001, that's about implementing information security policies within your organization, and some implementing some security standard. That will be the better solution that you can do against protecting this social engineering attack. So with that I will conclude my presentation. Thank you very much.
Thank you, Nawad, for that informative presentation. So now the presentation is open for question and answer. Any questions from the audience? Okay. Now I have a few questions. You said that when compared to the literature, when you access the literature, you found very few records on this social engineering attacks. So why is that? So does that mean the social engineering attacks are that much severe when compared to the other attacks? Is that so? So I think Mr. Naud is joining with Google Meet due to the restrictions, because he's abroad and he's joining with us. Okay, Naud, can you hear me? Okay, right. So you mentioned that. So when you search literature of the social engineering attacks, you found very few literature. So if I argue, so low literature means social engineering attacks are not that much severe when compared to the other security attacks. Is that the case?
That means like attacks happening from maybe like computer viruses, malware, ransomware kind of things, and then we have the social engineering attacks. So when it comes to the social engineering attacks, like it's like happening because of some, the attacker always tried to exploit the vulnerabilities remain in the human psychology or human mind. So it's like in that case we can't do anything, because we are not dealing with machines, some instruments or whatever the electrical things. It's dealing with your human psychology. And because of that thing, these kind of social engineering attacks are more they give more adverse consequences compared to this all other cyber attacks. So that's the thing.
Right. So as the mitigation approaches, so you said policy making, auditing, So that is, and also the biometrics. So when comes to the company-based mitigation approaches, policy making auditing is okay, and also the biometrics. And but my question is, so nowadays most of us are accessing social media, right? So as an individual, general public, so for them, this policy making, auditing, biometrics, so those are not practical. So when it comes to the general public, what will be the most applicable ways to get rid of these social engineering attacks?
I am so sorry for the inconvenience. I can't hear again. Karuna, I can't hear again. Can you once again ask, is it possible?
Now, can you hear me?
Yeah, now it's audible, yeah.
All right.
Yes,
So, as the mitigation approaches, you mentioned that. Hello. Now, can you hear me now? No. Yes. Okay, right. So, as the mitigation approaches, you mentioned policy making and auditing and also the biometrics, right?
Yes.
When it comes to the company-wise, so it's okay, policy making, auditing, biometrics. As you and me, we are accessing social media. For us, this policy making, auditing, and biometrics are not practical. So when it comes to the general public, what would be the best solutions to get rid of the social engineering attacks?
Now, did you get the answer, question?
I'm sorry, I'm like, I cannot hear the, I couldn't hear the last part, honestly.
So my question is, as the general public...
No, wait. Can you?
Sir, hello.
Can you hear?
Yes, I can hear now.
Okay.
Okay, now, so for the general public, so what are the possible mitigation approaches to get rid of social engineering attacks other than policy making, auditing, biometrics, and all that?
Okay, I assume the question that you are asking is what are the available, whatever the social engineering mitigation approaches apart from this technology and whatever this policy making, that kind of things, right?
Okay.
Yes.
Yeah, okay, so the thing is, like, based on the literature, I have reviewed all these, whatever the solutions, into two categories as policy making and then whatever the technological approaches. And I'm afraid, these are the only solutions that are available in terms of social engineering attack, because when it comes to social engineering attacks, you don't have any option, I would say. So you don't have any option because it's always try to exploit the human, vulnerabilities in the mind. Then that's how, it happens, because always when it's come to human being, they are too emotional, and whenever if someone maybe send in a mail, someone tried to send in a promotion mail saying that you have won this much amount of prizes, and so always because of your emotions, and as you are a human being, so you will you will fall into that, and then what will happen is you will eventually be the victim. So apart from these technological based solutions and whatever the policy based things, and educating people about these kind of attacks, so these are the only options that are available.
Okay, now, right. Thank you for the presentation and also the explanation.
Thank you for the presentation. The next one is there. Please wait.
Right, so we will move to the last presentation of the session, IoT Security Overview, Challenges and Countermeasures. The authors are N.N. Tilakaratna and N.R. Samrasinghe. The presenting author is N.N. Tilakaratna. Over to you.
Yes,
So this is regarding the abstract that we have submitted about IoT security overall overview, challenges and countermeasures. It's a kind of like a simple review. So if I may highlight the background of the research, so basically Internet of Things, so IoT refers to billions of devices, digital devices that is connected to the internet that can intercommunicate between between each other devices. So as per the time with these IoT devices, they are more vulnerable to cyber attacks than any others. Endpoint devices, maybe like a smartphone, laptops, because they are always, these devices, they actually always connect to the internet 24/7, and because of that reason, it is very easy for attackers to find out the devices using simple methods and compromise the devices, eventually endangering the critical infrastructure life of people. So this is becoming a huge problem for the time being in variety of smarter domains like smart cities, smart, smart medical hospitals, then transportation and surveillance, kind of all the smarter domains. So as I have obtained this information from the internet, as you guys can see, this is the statistics regard to the IoT security by the time of 2019. So the thing is, as for the time being, most critical infrastructure like a smart grid, power plants, all the things are powered by this IoT. And if something happens, what will happen is it will jeopardize the security of entire facility. That's the thing. So basically by doing this simple review, our main objective was to, by performing this thing, so examine the underlying security challenges pertain to the IoT architecture and examine the examine the categorized IoT vulnerabilities and outline methods for ensuring the safety of such IoT systems. So with regard to that IoT, commonly found IoT architecture, we will be categorizing these vulnerabilities, and we will be proposing countermeasures based on the literature, and that is for ensuring the safety of such IoT systems. So the methodology followed, what we have followed is like this. We have performed the keyword search using IoT security and IoT security, and we find out the papers in this index databases like Scopus, IEEE and Web of Science. So yeah, while performing this review, we found the author, some of the authors have mentioned like 3-layer architecture, and most of the authors have mentioned like there is a 4-layer architecture. So the most common architecture is like this 4-layer architecture, where in the bottom layer it is comprised of sensing layer, then we have the network layer, data processing layer, and the application layer. The sensing layer comprised of physical IoT devices, network layer comprised of networking components, and data processing layer, it actually acts as the intermediate layer between this application and the network layer. And application layer, it serves as the main layer that provide end user services for all its stakeholders. Like it will provide some facilities like smart medical health application, like smart city applications, kind of things. So I'm not going to go through all these things. Like then what we have done was, based on the architecture, we have categorized each of the security vulnerabilities and the attacks that target each layer. Like sensing layer, what happened is these these kind of attacks. So there's a hacker. They will go to they will go to physical. They will actually compromise the physical security of these devices at the sensing layer. And the networking layer, what happened is either they're gonna eavesdrop the whatever the message that will be transmitted or the communicating through the network layer, and they will try to compromise the message, or through the internet they will access to the internal network and they will compromise the networking components. So the data processing layer, it is actually much similar to the network layer, but mostly the attackers, they will try to compromise the data storage devices, maybe like cloud storage kind of things. And on the other side, the application layer, what happen is they will be actually coming through the like a front end of a particular smart solution. Maybe like if it is a web application, they will try to compromise the vulnerabilities in that web application, and they will eventually try to compromise the entire application. So that is basically how this security attacks target each of these layers, and it is found that I mean like uh, all four layers in this IoT architecture is vulnerable to most of these attacks. So then we have searched for the countermeasures that are available, like authentication and authorization. That means whenever users are accessing to the devices, it will be if we have deployed any authentication authorization methodology, that it will identify who is going to access and what access privileges that we will be granting to that person. Then the data encryption and integrity, that means all the sensitive information like medical records and kind of things are needed to be encrypted using this data encryption. And data minimization, that means only collecting the necessary personal information that is deemed necessary. And continuous monitoring and reporting, that means maybe using by deploying network operations center and kind of things, we can continuously monitor all the networking facilities and the critical IoT infrastructure all the time. Maybe we can incorporate this real-time artificial intelligence kind of things, that is also possible. And using tamper resistant hardware, that means the hardware devices must designed in a way that is hard really unbreakable by the physical security attacks. So it's called tamper resistant, and the following the best practices like daily updating whatever your devices kind of things. Then having awareness session for end users in the organization. These are the kind of best practices. And divide the network into segment. That means if you divided your entire organization network into separate segment, what happens is then the attacker, if attacker performs any attack, that will be limited to a one such network, and they might be not able to escalate their attacks to all other networks, so that will be limited to that particular portion. So it is better than compromising the entire network at all the time. So these are the things that we have actually found through a simple review. And yeah, the final conclusion. So as we have found that based on our review, there are security attacks that target almost every layer in the IoT infrastructure. And the threat rate imminent, because whenever you have the device that is connected to the internet, the attacker can anyhow, if there are some security loophole, they can found out and they can compromise that vulnerability. Hence countermeasures needed to be taken, so that is a must. So at least one or two countermeasures can be taken. So and based on your organization, like IoT technologies, based on your organization and your employee needs and things, so it's better to keep them aware of such things. So it will be better for your organization to have a better security against this cyber security vulnerabilities. So that's about our presentation. Thank you.
Thank you, Mr. Nawad. So let's move on to the discussion. So any questions from the audience? Okay, again, I have a few questions. Mr. Nawad, can you hear me? Okay, so you mentioned about 4 layers, the common 4 layers, the application, data network, and the physical layer. So I'm asking, out of all, what is more vulnerable and why?
What is it?
Out of the 4 layers, what is more vulnerable and why?
Out of 4 layers, what is the more vulnerable layer?
Okay, out of 4 layers, from based on the literature and from my experience also, I would say that the outermost application layer is the most vulnerable layer, because it is the actual the layer that is actually dealing with the end users. And because of it is the front facing layer of it's the front facing layer of whatever the IoT solution is, it constitutes the front end of the solution. So it is the most vulnerable layer when it's come to like 4-layer architecture.
So, okay, right. You are talking about access control attacks and physical security attacks. So, at the recovery stage, out of these two attacks, what is faster to recover?
Can you say it again?
So, there are two types of attacks.
There are two types of attack.
Access control attacks and physical security attacks.
Access control attack and security control attacks.
So, out of these two attacks, when it comes to the recovery stage, what is faster to recover?
Out of two attacks, when recovery stage, what is the
Fastest.
Faster
to recover.
Fastest recovery of these two.
So I got the point. So it actually depends on the situation. In terms of access control attacks, let's say the attacker may have breached the computer network and access to some vulnerable files. So the files may contain some really confidential information, and he already got whatever the information included that in files. And now when it's come to forensics, they are trying to recover recover what has happened to going through the log files. And so I would say also when it's come to physical security also, so it actually depends on the situation and the context. Whatever the organization size, then whatever the magnitude of the attacks and the knowledge of whatever the attacker who is trying to compromise all these physical security or access control things. That's why. So it actually depends on the context, I would say.
Okay, now thank you very much. And
thank you.
So this marks the end of the technical session 4 and also the end of whole technical session. So again, thank you very much all the presenters and congratulate all for their presentations, and let's wish we'll meet again on the next annual research symposium technical session. Thank you. Thank you very much.
Thank you very much, Dr. Chaturika. With that, the annual research symposium of the Faculty of Technology, University of Colombo is now concluding. Thank you very much for all the presenters today. Your research work added glamour to the symposium. Now the proceedings of the 6th annual research symposium of the Faculty of Technology, University of Colombo is officially concluded. Thank you very much indeed for your presence and your valuable contribution. I hope you have spent a productive time with us. Have a pleasant day.